I'll be starting off with explaining the cultivation of the algae and its isolation, and then upscaling the isolates from the lab scale to the large scale, and then further its biochemical characterization for the lipids and characterized by PCMS and HPLC. So this will help the PhD and the PhD scholars and the students who are participating in the workshop to use these techniques in their daily life. So coming to the why algae and why not the other crops besides algae, it's because the algae, they have very unique functions. The very first one, they are very fast growing and they yield higher productivity. And moving towards the second, they are able to use the waste and the salt water. For example, we can cultivate the algae in the coastal areas where the seawater is abundant, and the algae can be, and that water can be utilized for growing the algae. Since the seawater contains many nutrients which are essential for the growth of algae, and so it may help to reduce the cost of the nutrients which we are preparing using the commercial media. And moving towards the very important aspect that the algae, they are very non-toxic and they are biodegradable, and they have huge application in the biofuels, which includes the jet fuel, ethanol, gasoline, and diesel. And also they have a huge application in the nutraceutical markets. In the value, they produce multiple products, as in the yesterday presentation we have seen that they are good producer for phycocyanin. They produce the beta-carotene, astaxanthin, and other value-added products. And they are further able to recycle the carbon dioxide. And very important that the algae do not need any arable land. So they can be cultivated anywhere. We need just a small area, a pond, where we can dig and just cultivate the algae. So moving further, so before we start off with the algae, we need to select the habitats for the microalgae. So for understanding the habitats, we need to first understand and mimic the environmental condition. So the very initial step when we go for collecting the algae from the water samples for algae, we need to focus what we are targeting. So if we are going for the soil, that is the terrestrial algae, we need to usually look at the surface soil from the diverse habitat and collect the samples from there. And from the water bodies, we need to take some amount of water from different regions and from the diverse habitat, and we can collect those samples. And also there are few floating microalgae which we all can see in the ponds, static ponds and the lakes. They are growing at the surface, so we can collect those microalgae very easily by using a mesh or a muslin cloth and a sieve. So once you have collected the samples, they are collected in a vial which contain any enriched media. It can be a basal media or a media containing nitrogen, phosphorus, and magnesium. So if we talk about, there are many different habitats. So the very first one, if we target the coastal marine algae, so the very important thing while you are collecting the samples from the coastal area, we need to focus on the temperature, because in the marine area the temperature sometimes is very high and there are some areas where it is very low. And second, the salinity is the very important thing, because the marine algae, they generally grow at a salinity of 36 gram per liter, which is and below that the strain cannot survive. So while collecting the sample, you need to when you're collecting the samples, you need to measure the temperature and the salinity, so that while you are bringing the samples to the lab back, you can adapt those conditions in the lab, so it would be easier for the algae to grow easily. And if we move further towards the oceanic open seas, then there are also challenges, the water quality, because there the metal toxicity is very high. For example, heavy metals like lead, arsenic, zinc, so they are present in that water, which may be harmful which may be good for growing the other organism, but at the same time it can be harmful for the algal. So you need to measure the quality of the water which you are collecting. And moving further towards the fresh water, they are a bit easy to grow, but again they are less sensitive to temperature, so not an issue. But with the freshwater algae, pH is a major concern. So the pH should not go below 5. It should not be acidic. And moving further towards the polar and the snow algae, they are sensitive to the warmer temperature, and they also need very special culture media to grow. And last but not the least, which are the terrestrial and the soil algae, they are the environmental factors, they are least important, so they can be easily grown while bringing the samples to the lab. So once we have collected the habitat, the water samples from the algae, we bring them to the lab, and then we try to isolate them. So before isolating, we need to eliminate the contaminants which are there in the algae. So there will be many prevailing contaminants along with the algae, which can be bacteria, fungus, etc. So they can outcompete with the other target species. So very simple step before we bring the samples to the lab, we need to pre-filter the samples immediately upon collection so that it is very effective for removing the unwanted organism. And also the another technique is we can do the dilution followed by the streaking in the agar plate. And also we can do this continuous subculturing in order to get the single colony. So for the isolation, we follow very basic steps, which are the spread plating and the serial dilution. So we can do the spread plating, streaking across the plates in agar media of a specific, for example, if it is a freshwater, we use the freshwater media, and for the marine, we use the marine media. And second, we follow the serial dilution method, which is a very effective way for the organism that are rather abundant in the samples. For example, we can grow the cultures in the lab and serially dilute up to 10 to the power 6 dilution. So if we do the dilution to the 10 to the power 6 and 7, at that time maybe the other contaminants, they are eliminated in the early dilution. So we go for the larger dilution so that we get the pure colonies of the algae. And then further proceeding after the isolation, we do the purification. So once we have obtained the uni-algal isolate, we treat the samples with the bioleaching agents followed by plating them on a agar media. So we do extensive also washing with one or the more antibiotics. But here the problem is with the antibiotics, they are very specific to the specific strains. So if we say one strain can sustain the streptomycin to a particular concentration, maybe another strain may not survive in that condition, and it also inhibits the growth of the algae. So my suggestion would be that instead of using the antibiotics, if we go for any bioleaching or the detergents which can be used for the purification of the algal isolates. So once we have done the purification, we do the these are few of the pictures which clearly indicates that after we have collected, we have isolated on the specific agar plates. So if we look at the left image, it shows the autotrophic cultures, and we are getting the pure colonies of the algal isolate which contain no carbon source. And on the right hand side, we have the heterotrophic organisms. So they are growing in presence of glucose. And below you can see on the left hand side, we have purified the cultures on the agar plate, on the nutrient agar plate, which is the enriched media for the bacteria. So you can just pick the colony from the algal agar media and then streak on the nutrient agar in order to see whether the algae is growing without bacteria or bacteria is producing more. So as you can clearly see that we were able to attain the pure colonies of the algae in the nutrient agar media. So further, once we have done the isolation and decontamination, we proceed with the enriching the culture. So enriching the cultures means once we have obtained the uni-algal isolates, we use the nutrients, we provide the nutrients for algae to grow. So the common enriching nutrients which are there for growing the algae are, you can add the soil water extracts. There are several macronutrients. The very common are the nitrates, ammonium, phosphates, calcium, magnesium, and we use few in the few medias the trace elements. So for establishing a unialgal culture, we need to take care of the few parameters which are very crucial for growing the algae. So coming to the temperature, so temperature is very critical factor for the algal strains. So if you have collected the algae from the marine habitat, then maybe 20 degree, 19 degree, 16 degree would be good for the marine algae strains to grow. But it again depend on the location from where you have collected. So you need to actually understand first the location and then mimic the same in the lab conditions. So light is not that much issue for freshwater and marine. So the optimum light can be good for growing these cultures. Again, pH is very important factor for freshwater and marine. For freshwater, generally we 6 to 7 pH is good for growing this strain. But if we talk about the marine isolates, they grow at a pH of 8 or 8.5. And the most important thing is the mixing, like whether the culture should be grown under static or they should be grown under this shaking condition. So if you have collected from a pond, then that algae may not need a shaking. But if you have collected from the streams or the tides, then yes, that strain might need to be put under the shaking condition. So once we have established the unialgal cultures, we then further move to choose a culture media. So for culture media, nutrient concentration is very crucial, because if some nutrients are oxidized or they are otherwise they are gradually altered, especially during the light illumination. So also the quality and purity of the agar media which we are using also play a significant role on the growth of the algae. So once we have established the media, we have enriched the media. So further, there are few frequently asked questions which needs to be answered. So the very first one is, why algal species grow in initial stages of isolation but then die after 1 or 2 more transfer to the fresh media? So it may be due to the lack of the culture media which we are using, and also the time is very critical factor when isolating an algae, because if you have collected the samples and you have kept in the lab for 2 to 3 days and then you think that you can isolate the algae, then it's become really tough. And then is the enrichment, which should be, so if once you have enriched the culture, you need to observe it daily in order to check whether your algae is growing in that media. If not, then you need to manipulate the nutrient conditions. So moving towards the question 2, which is how much enriching material should be added. So the answer is not that much, but yes, there are few algae which has the ability to grow at high CO2 concentration. So for them, and culture enrich should be rated with 1 to 5%, which is an effective way for the species to grow under high CO2 tolerance. And also there are many different types of selective cultures which can be designed. For example, you can use high or low temperature, light, salinity and pH according to the habitat from where you have collected the isolates. So coming to the last question, which is whether a particular strain would be best to be maintained for longer period in liquid media. So yes, the solid media are preferred to be for the storing the algal isolates, and exenic strain are sometimes best to be cultured in a media which is rich in the vitamins B1 and B12, and they can be kept under the light and dark photo period for further maintenance of the cultures. But sometimes inappropriate light is to dark phase photo period may lead to the deformities in the, it may inhibit the growth of the algae. So once we have now enriched the media, we further go towards growing these algae and study its growth pattern. So that study I'll be showing in this clip. So what is this? This is a multicultivator where we grow the algal strains, and in this multicultivator we have the 8 panels which can take at the same time 8 different species, and they can grow at 8 different temperatures, 8 different light regime, and 8 different photo periods. So you can use this equipment for bioprospecting of the algae. So I'll just play this 1 minute. We cultivate the algae in this mini photobioreactor, which contains the 8 growth panels. And so once we have this instrument also helps in generating the real-time data of the optical density at 680 nanometer. So we extract those data from this multi-cultivator. So after then we can see the multiple pattern of growth. So the samples are collected from freshwater species and lakes and the chemical polluted areas and the terrestrial algae. We can clearly see that under same control condition of 120 microangstroms per meter square of second light intensity, 16:8 hours photo period at 25 degrees Celsius, these strains behave differently. For example, this Canidesmus collected from the same habitat, they are also behaving differently. So we can't say that the samples for example, we have collected the samples from Vishakhapatnam, and that samples contain 4-5 Scalidesmus strains. So that doesn't mean that it will respond similarly. So this Scalidesmus, so the strains collected from different habitat may respond differently under different conditions. So after we have done the we do the growth kinetics. We further move to now characterize these strains in terms of the potential of producing the lipids and the carotenoids. So after we have done the growth studies, we then use the dye, which is body piece. We stain the cells with the body piece, and we visualize it under the confocal microscope. So we can clearly see that the lipids body in the green on the right hand side, the few are present on the cell membrane, and there are few lipids body which are present intracellularly. And on the right hand side, we can see the green droplets show the lipid containing, and the reddish orange droplet shows the chlorophyll. So this is the qualitative analysis we do through the confocal microscope in order to see whether the cells at different days of harvest has accumulated lipid or not. So before we proceed with the total lipid analysis and the GCMS profile, we first do the staining with the PODP, and then further we move for the quantitative analysis of the lipids. So we do the extraction of the crude lipid first. So we take the dry powder of the microalgae and we do the solvent extraction. So generally the most preferred method for extraction of lipids from algae is used by Bly and Dyer. So once we have done the extraction, we filter the samples through 0.22 micron filter and transfer the same to a glass vial or a beaker, and the portion of lipids are extracted and solvent evaporated at 40 to 50 degree, and the lipid residue is further dried, and we calculate the total lipid content with the formula as mentioned over here. And once the lipid are dried, we then derivatize those lipid in order to be analyzed through GCMS, because fatty acids, they are non-volatile. We convert to the volatile by derivatizing it and then analyzing it through GCMS, and this was discussed in detail yesterday in the other presentations. So this is a method we quantify the lipids. And moving towards the quantification of the carotenoids. So the carotenoids we analyze through HPLC. So once you have extracted the carotenoids using the solvents, organic solvents, we quantify and we do the qualitative and quantitative analysis through UV with spectrophotometer and HPLC. So the most important thing while you are carrying out the extraction of the carotenoids is that they degrade very easily. So if you are using any carotenoid standards, you need to make sure you take care of the temperature and light, because they are very light sensitive, and again if they are kept at higher temperature, it will degrade. So you need to take all this precaution while you are carrying out the analysis of the extraction of the carotenoids. And for HPLC analysis, again the flow rate, temperature, running solvent, and the injection volume, they are very important factors. So for HPLC analysis, it depends on the focus which you have, like which carotenoids you are extracting. So you need to take care of these parameters. So the HPLC analysis will provide you like a different carotenoids which are present in your sample. So but if you want to further isolate the particular carotenoid, you need to further go for the column chromatography and then get the single fraction and again analyze through HPLC. So this is the overview of Fico prospecting, how we do, which I have explained. So I'll just brief you. We collect the natural culture samples and give the nutrient source, and then we upscale. We grow them in a lab, then upscale to the large raceway ponds, and then we further harvest those algae. We dry them to the solvent extraction. So we can using the organic solvent, and the another method is the greener approach, where we can use the supercritical fruit extraction and the biomass which is left over can be further used for as a fertilizer, or it can use for aqua feed for cattle. And further the biomass is bioprocessed for lipids, uh, which include the omega-3 fatty acid and the valuable carotenoids. So then further moving towards how now we have attained like the strains which have good potential of lipids and the carotenoid. So now we further move how we can enhance these. We can further enhance these metabolites and the lipids. So there are many factors which are used, rate limiting factors which are used for scaling up these metabolites. So for example, under lipids, there are many reports which says that when we use low temperature, there are increased reactive oxygen species which are generated, and due to that it's lower its growth rate, but it increases the lipid content. And there are many studies which shows the effect of salts, nitrogen starvation, that it can help in increasing the PUFA productivity. But this is very generic, like the reports, they have mentioned that you can use the low temperature, you can use high light, but it all depends what is your target. If you want to go for extracting the multiple products out of algae, then you need to use the combination of multiple stress factor in order to achieve both the lipids and the carotenoids at the same time. So once we have achieved, so there are reports which are delivering the single products. So this is taken from one of the reference paper where they have mentioned there are species which are prevailing, for example chlorella. So they have targeted the product as biomass and they are growing in the open ponds and the glass tube photobioreactors. And there is another very commercialized isolate which is Hematococcus prevalis, known for astaxanthin. So it has good application in feed additive, but they are again growing this in an open pond. So these are all strains which are delivering only the single product. But for the microalgae to be commercialized at a large scale, we need to look at the multiple products. So these microalgae, there are some commercial exploited microalgal strains. For example, chlorella, they are well known for lipids and carotenoid, and this was discussed yesterday, which is spirulina. Another name is Arthrospira plantensis, and it is a good source for phycobilly Portococcus prona, again a good content of lipid. So it is just if you extract 55% or greater than 50% of lipid, then you cannot extract the other metabolites, because this is a strain which contain the higher amount of lipids. Then we have Schizochytrium, a good source of DHA. Then Pheidodactylum, which is a good source of eicosapentaenoic acid, and so on and so forth. Dunalila for beta carotenes, Candidesmus elmarensis, a good source for lutein, and Tetraselmis for omega-3. And Hematococcus, a good source for astaxanthin. So now coming towards the market potential of these nutraceuticals and metabolites. So if we look at the market potential, they have a very huge market, which is around 200 USD million for astaxanthin, and they have a very good application, which is in natural. They act as a natural antioxidant and they help in stress management. And there is another very good eicosapentaenoic acid, which is omega-3, and it is found in lot many species. And Nanochloropsis opulata has a good amount of EPA content, and that strain is also very well exploited, and it has a market of 300 USD millions. And so on. There are many products which single products which people are delivering, and there are many industries, key players which are there. And EID TERI nutraceutical, they are producing the phycocyanins, and they are growing them in the open pond, and they have a huge market potential of 50 USD million. So, but since they since due to this high market potential, why the algae is still not commercialized? So the answer is that there are a lot many scale-up challenges still prevailing with this technology to be commercialized. So the scale-up challenges are that the select the condition of mass cultivation it depends on the habitats from where you have collected the isolation. So when we are scaling the cultures to the outdoor, we need to actually look into the strain from where it was collected. For example, if there the light was we have collected from the shores, deep shores, then you need to say what is the light. So generally the outdoor reactor system, they have like the natural sunlight is there. So you need to mimic that environment. You need to put some artificial light also with the natural light. So we can try this out. And also the scale-up challenges include, for example, if you have collected from ponds, then you don't need to put the impellers for rotating that algae. And but for of course for the samples collected from streams, you need to have the movement in that samples. And last but not the least, the selection of right media and photoperiods are the most important factors while growing these strains. So from the scale-up challenges, we have significant commercialization challenges also, which are still prevailing, and we need to address them. So there are two overcoming challenges, which are reducing the cost of production and they are affordable and scalable algal biomass production. So one is the non-efficient nutrient recycle for the production, and also the current commercial technologies, they are designed for production of high value products rather than for delivering the single product. And also the novel species, we need to target the species which has the good potential of producing these metabolites and lipids. And of course, the downstream processing is also a great challenge because harvesting is a major issue. And the challenges in harvesting are that there are many challenges in harvested, but nowadays many reports and people, they are using the wet harvesting, or they are using the extraction of lipids by binding precursors, and they are using flocculants. And there also there are reports suggesting the gradient centrifugation without dewatering and leaching out the metabolites. So at the last, this I want to just show the common facility for the algal work at TERI, which we are using for characterization of the lipids and the metabolites. So I would be happy if anybody wants to use this facility, they can contact us and we'll be more than happy to address their any concerns or issues. At the last, I would like to thank Dr. Alok for mentoring throughout this journey, and I would like Suchitra, who is working as a project associate in the one of the project, a DBT funded project for enhancing the metabolites in the outdoor reactor system, and Priyanshu Jain for working on the enhancement of the omega-3 fatty acids. So thank you very much. So now I'll play the demo clip, and then we'll end up with this session. Thank you.
We are here demonstrating a process for lab-scale protocol for isolation and multiplication of algae from small-scale to large-scale cultivation. The utmost important thing is that before we proceed with any work related to algae isolation, all the necessary requirements of glassware, plasticwares need to be properly sterilized and kept under UV for 20 minutes. So the samples collected from diverse habitat, that is freshwater and marine, are harvested at low RPM centrifugation and inoculated into the respective media, that is bowl spatial media for freshwater and F/2 for marine. The cultures are kept under optimum growth condition of 120 micro-Einstein per meter square per second of light intensity, 25 degrees Celsius temperature, 16:8 hours photo period in shaking. So after 7 to 10 days, when the growth seems to appear, the cultures are serially diluted up to 10 to the power 5 and 6 dilution in order to get the pure colonies free of other contaminants. Further, the 10 to the power 5 and 6 dilution are plated on agar plate of specific media and kept under controlled condition in algatron incubator at 120 micro-Einstein per meter square per second of light intensity, 25 degree Celsius temperature, 16:8 hours photo period until the colony appears. So once colonies appear, single colony is picked and inoculated into 2 ml of media broth and kept under the controlled conditions until the colony appears to grow. So further, the cultures from 2 ml is upscaled to 70 ml for growth kinetic studies in Multicultivator for bioprospecting. Further, the cells were recovered by centrifugation and washed 3 times with water and then inoculated at an initial cell count of 3 into 10 to the power 6 cells per ml with the aeration rate of 0.2 ml per minute at 25 degrees Celsius temperature, 16:8 hours photo period at 120 micro-Einstein per meter square per second of light intensity. So the real-time data for monitoring of the algal growth is measured at optical density of 680 nanometer and 750 nanometer. So the cultures are further upscaled to 5 liter and then to 50 liter. So from 15 liter, the cultures are then transferred to the raceway ponds. Before transferring the cultures, we need to take care that the raceway ponds need to be cleaned properly with water and then subsequently with ethanol. So after cleaning, the nutrients are added to the pond and the volume is marked up to the scale which we want to cultivate. So further, the culture with 10 to 15% of the starter inoculum is inoculated in the outdoor raceway pond, and the optical density and pH are continuously monitored on the daily basis. We need to make sure the OD should not go below 0.2 in order to prevent the cultures from dying. So at different phase of exponential and stationary phase, the cultures are harvested and the flocculants are added to the harvested material. So after overnight settling, the cultures from the bottom is concentrated and harvested for further use in characterization of biomass for lipids, protein and other bioactive molecules.
So I would like to thank the team for preparing this demonstration clip. I would like to give a special thank to Suchitra, Priyanshu, Dr. Shovan, and Gurpreet. Thank you.
Thank you, Dr. Amrit. We have some questions from the participants. So we'll take the first question, which is by Kushal Mehta. And the question is, can each algal strain be grown on solid medium?
Yes, the algal strains can be grown on the solid medium, but the thing is you need to first isolate and purify the algae, and then you can grow it on the solid medium. Okay. So if you have some bacteria which is competing with the algae, so you need to first purify that culture and then inoculate. So I don't think that it should be a problem growing in a solid medium.
The next question is from Pawan, who is saying, ma'am, what kind of difficulties we face in scale-up of microalgae from lab scale to pilot scale or industry scale for bioethanol production?
For bioethanol, I can't comment, but yes, for upscaling the cultures from the small scale to the lab scale for the algae production, for the biofuel production, yes, there are challenges, because when you are growing the algal strains in the lab scale, they are all growing in the artificial light condition and all artificial condition of temperature. But yes, when you transfer them to the outdoor conditions, then definitely the strain, they come into the stress and they get a shock. So what you need to do is you need to first acclimatize your isolate in the lab, which is there in the lab condition, to the condition where you want to cultivate. So you need to transfer that 15-liter can to the outdoor reactor system in order to acclimatize your strain to the outdoor condition, and then you can transfer into the 300-liter or 1000-liter of reactor systems.
So we don't have any other questions. So thank you, Dr. Amrit. So it is well mentioned by the presentation that microalgae isolation, culturing, and prospecting. We hope that the knowledge shared will lead us to a product holding commercial importance. Thank you so much. I would like to request Dr. Shawan to present his lecture now. Dr. Shawn Mundel, a Ramalinga Swamy Fellow at the TERI Institute, has made substantial contribution to the field of algal biotechnology. He received his PhD from the IIT Kharagpur, where he worked on algal physiology biodiesel production. Prior to joining TERI, Mundel held associate research scientist position at New Mexico Consortium and postdoctoral research positions at Oak Ridge National Laboratory, the University of Michigan, and the University of California, San Diego, USA. During his research tenure in USA, he demonstrated how fundamental principles of ecology can be used in algal biofuel production. His research helped to generate an environmentally sustainable route for improving algal productivity and biofuel yield. So I would request Dr. Shawn Mundel to start with his presentation, please.
Thanks, Dr. Ruchi. So, very good morning, everyone. So today I will talk about sustainable microalgae farming for biofuel and other commodities. So before going to this topic, so I want to tell something about the recent history of microalgal research. So the top graph, if you look, so it's showing the number of publications on microalgae in each year. So if you look at this red line, it's showing the real values of like how many publications from the microalgae each year. but it's increased after 2005 and 6. So the question is like, why people is loving microalgae from that era? Like, what's the reason like that much of publication? Why increase after 2005? So I try to relate those things with the crude oil price in the international market. So if you come in the bottom graph here, so you can show it's showing like almost 70 years data of like how the crude oil price changes over time. So it's like below $20 to it goes like above $120 over time. And if you see, like I was talking about 2005, just like an example here. So what was the price at that time? So if you see, like the oil price was at that time increasing so high, and there is a push from the government and other agency, like, yeah, we need to find an alternative, and where that microalgae that can play a huge role. And at that time, the microalgal research actually started a huge, actually, I would say that's a huge start. But if you see, like in the past, like in 1970s, so oil price also goes high above $100 per barrel. And there is a aquatic species program, that is a project. It was led by National Renewable Energy Laboratory in US, and it was funded by Department of Energy. It was started in 1970s, and when the oil price increased so high, and they try to find like the algal species if they can use for the biofuel production. And I would say, like, if you see the report from the aquatic species program, that was a huge success, I would say. Like, they found a lot of species in that time. They're having a lot of lipid content. They find like some genetic diversity among the species, and also they find like what are the genetic mechanism for the lipid production in the algae. At that time, because despite of the huge success of this project, still the project was ended in 1996 because the oil price is dropped down at that time. So the price is also a factor, like oil price for the biofuel research. But I'm very happy to see like the oil price is decreasing at that time in the last few years, but still the microalgal research is going on and a lot of publication is coming out. So, but if you look like the focus of the research of microalgae, like in the publication, or if you look at the agenda of any international conference, like ABO, like Algal Biomass Organization, they organize every year. So if you look at the agenda, like from 2010 to now, so if you can see like the agenda is going to change, like they are much more focusing not only on the biofuel, like other derivatives, like other products, like the bioplastics, they're currently thinking, like any other products, like from the last days, actually we're discussing a lot of other products. So they're taking a lot of pages on the your agenda, but it's not like too much biofuel. But I personally believe that algal biofuel that will comes again and comes as a main focus because of their potential. And when the oil price will be going high, and so that's the current status, I would say. And if you look at the publication from each country, yes, the maximum publication is coming from the USA, followed by China, and India is doing great. Like it is just like followed by India, USA, and China, and Australia, and some other countries. If you look, so they are also performing very well in the algal research. And but I think like why this microalgal biofuel? Just like a few minutes back, Dr. Ramrit was talking like, yeah, why microalgae important? But I have been working from my PhD on the biofuel, microalgal biofuel, and people always ask me, hey, when you'll give the biofuel from algae? But I always try to remember the people just think like the petroleum that you're currently using your car right now, they are mostly fossil algae. They come from algae. But it took a long time, like a million years to store those products. But since industrial evolution, what happened? We're using so fast, so we have like a less than 100 year left, like if we could use in the same way. So we don't have like those storage products. So what would be the next? But you could think, like just imagine, like 300 million year storage, we are just burning just in 8 to 10 human generation. So we are just enjoying our cheapest source currently. We're not producing anything. So what may come, like, so the microalgae can play here a big role because they are fast growers. So people are domesticating algae and making biofuel in real time. So that is biofuel. So because if you see in this graph, the productivity of the algal oil, so it is like 2 to 3 times at least from the high oil crop, like your oil palm, if you compare. So, and it is possible to, that is your real data, like from the industry, like how much possible oil production. But there is a lot more possibility. But I am not talking too much about like those, there are other advantages, so it's already discussed. And I would say one thing, like this microalgal biofuel, it's not a scientific hypothesis, it's a reality. So the green card, if you see on the top, it's the Algiers, it's already run from Los Angeles to New York, like it is more than 4,500 kilometers by using microalgal oil. So I would say like we have shown like it is possibility, but still there is a lot of challenge, of course. As earlier discussed, like yeah, the cost, always people ask about it. And how could we reduce the cost? Like we could, if we increase the productivity, so there is a possibility for reducing the cost. And at the same time, the crop protection, that is a huge challenge. If you see on the bottom graph here from the sapphire energy, so when the pond is infected by the fungus, it iterates, and just like a matter of few days, 2-3 days, your pond will be gone. So not only this pond will be gone, like the whole facility, it's very hard to maintain at that time. And if such kinds of attacks comes in, and if you cannot control, and if you need to shut down your facility for just like whole things, just to clean up, so your revenue that will be reduced 60% in year, just in one shutdown, if you need to shut down everything. So it's a huge cost. So we need to think about how could we protect those things. And also, as Dr. Amit told, like harvesting, that's a huge challenge. Because like in normal system, like in our, when you'll be growing in outside in our tropic condition, like 1 gram per liter, that is the common values, like you can get that biomass. That is your 0.1% biomass. You have to collect it from the huge amount of water bodies. So that is a huge challenge. And also like this water supply, and also like nutrients demands like nitrogen, phosphorus, that is the major nutrients you need to add, like, but we have like the limited phosphorus in our world. So how could we do those things? So that is also a huge challenge. And people are thinking like the wastewater, yeah, of course, that can be a big role. So I will talk in details. But today in my talk, I will mostly focus on this increased productivity and the crop protection, not all those details. So how do you increase the productivity? Yeah, so people think like, yeah, so if you add more nutrients, there is possibility, or like taking some good quality species, that is also. But I will be telling something different, like how Charles Darwin's theory we could use like for the biofuel productions. Charles Darwin, more than a century ago, he talked about the division of labor, like division of labors between species that can enhance your total stability of a community. So just like in this picture, like if you perform like the different function, yeah, so our community will be much more stable. And this theory in modern time, it comes to Hutchinson niche partitioning theory. Like each species occupy a different niche, that means their needs like the different requirement. Just for an example here, the trees are growing, they're in the different root depth. So if they're extracting the nutrients from one layer, other species taking from the other layer, or at the same time, one species taking more light, one are going at the shading, so there will be less light. So if they love the less light, so if that kinds of combination, that will be possible. So at that time, so so all the species can grow together and will get much more productivity. But algae doesn't have any root system. We know they don't have like the cryptogams. But they have the preference for different things. Like they have, just like an example here, we took 5 different microalgae, Navacula, that is the diatoms, and we took 4 different green algae, that is the Chlamydomonas, Tetrasalvis, Rhaphidosalis, and Pseudosmus. They are well known for the biofuel production. And we measured their ammonium uptake rate, their urea uptake rate, and nitrate uptake rate. And we presented those data here. And where our hypothesis is there, like those species are in close together, and if you have all those nutrients, they will compete for the same nutrients. But if you grow, like just an example, if you grow like the Navacula with the Chlamydomonas, what will happen? They like the same kinds of nutrients, the nitrogen source, so they will compete for the nutrients. But if you grow like your Navacula with the Rhaphidosalis, so what will happen? So they are in the distantly. So one will take like urea, another will take like the ammonium. So at that time there will be a chance of like your higher productivity. So that is our hypothesis. And to test this hypothesis, we took all the 5 species, we grew them separately, and we just combined like the 2 species culture. So in 5 we'll get like 10 different kinds of 2 species culture. We called it as a polyculture, and the first one is a single species we called monoculture, and we mixed all 5. So we're having like the 16 different kinds of species combinations, and we grow all 16 species communities separately in 4 different nitrogen sources, that is nitrate, ammonia, urea, and we like in the mixture of 3 nutrients, keeping the all nitrogen concentration same, and we call it as a simulated wastewater, because like in the wastewater, like they are having possible like the multiple kinds of sources of your nutrients. So that's why, and we grow like multiple periods of time, and we measure like their growth. You can see it here. So I'm just showing like we grow like the multiple species, but we are showing the average of their production like for each species richness. Most of the species don't like this higher concentration of the ammonia, but they like the nitrate. But here I wanted to show here, if you look at the biomass production over the species richness, what is happening, if you increase your species richness, the production is going high. So that is the main thing I want to show it here. But at the same time, if you look at the wastewater simulated, the blue one, dark blue one, so if you look at the loop and the slope of the curve, at that time, that is much more higher as compared to others. So that is expected. Like in wastewater simulated, we have like the multiple kinds of nutrients, and we are growing on multiple species. They are taking all those kinds of nutrients, and we are getting much more productivity. So instead of growing like the single species, so just like here, we got like almost 25 to 40% increase of your biomass production, just increasing your species richness. So that's the interesting thing. But if you look most closely, like which combination they are giving better, so we try to compare with these things with the monoculture. So how do you compare these things? Like we can compare like the different way, but I am showing it's one type here. It's called overyielding in ecology. So just an example, like species B, when you are growing, they are giving like 120 milligram per liter. When you are growing E species, it's giving like 82 milligram per liter. So that's what is the mean of these 2 species? That is 105 milligram per liter. But when you will be growing the species B together, so you are getting 161 milligram per liter. So that means as compared to your monoculture, like so it is 56 milligram per liter higher. So we put that 56, this value, in the right hand side of the graph, and in the 0 line above, that means your positive overyielding. Similarly, there is negative overyielding. I am showing in the bottom graph, like here, like B is 128 and C is 112. So that is the mean is 120 here. But if you're growing BC, we are getting less. How much? That is 27. So it's negative. So it's going to the bottom of this 0 value. So all these things like the ammonium, nitrate, urea, and the simulated wastewater, we are showing those values here. And we saw like in simulated wastewater where all those kinds of nitrogen sources are there. So at that time we are getting much more of the polycultures showing their positive overyielding. But the question is coming like, yeah, the polyculture why not always they are showing your high biomass yield. So we try to correlate those things like in the next graph. So if you see, so that is the same graph earlier. I used it like this, how their affinity. So we converted this graph, three-dimensional plot into two-dimensional plot using the polycomponent analysis. That's showing this correlations. And here, like this, you can see like there are species like this, all where they're in the same way. And the axis in the PCA1 that indicates like how they are liking the same things like these nitrogen sources, like species A and C, Rhapidoscelis and the diatoms, they are in the distantly related. That means like if you take this distance, they don't like the same kinds of nutrients. They're different. So we are putting those PCA1 axis values, the difference in the PCA for each species combinations, that is here in this x-axis, in the next graph, on the right hand side. And we also use the productivity that is on the y-axis. And we see that when our difference is increasing between the species, because their affinity is different at that time, we are getting much more productivity. So you need to think about it like when you're making the consortium, not like the random inclusion. So what would be their liking? And in that way, actually, that need to be complementary. Otherwise, they can compete for the nutrients, and you cannot get much more productivity. So that's you think about it like before making any consortium. And another interesting thing also that is important for the any algal biomass production, that is your nutrients use efficiency, like how much your nutrients is using and how much biomass you are getting from that. So if nutrients use efficiency is higher, means you're much more economical in that sense. And when you'll be growing the polyculture, we have seen like actually nutrients use efficiency much more higher as compared to your monoculture system. So the two way you will keep benefited here, that is first increasing the productivity, as I have shown, like and at the same time you could reduce your nutrients cost. And I'll be talking about the third thing, like the crop protection. Yes, like you can be benefited from the growing your polyculture, because like if you grow like multiple species, if one insect or pest come, they are specific for a particular algae, so the others can take over, just like the portfolio effect, like when you're investing money in the share market. Don't invest like in a one share. So like people are doing like the multiple share, like something's like goes out and something fall down, so other can take care. So your overall benefit would be like stable. And biofuel industry always think about like the algal industry think about, I would say like the stability. People think about like, yeah, we can give them species, they can give you high productivity, but if they crash, so your loss will be much more than that. So that's this portfolio effect you have to think about. And where this polyculture, that's the sustainable way, I would say, thing to grow in the outside also. Because like when you'll be growing those things, like in the inside, it's not a huge problem, but in axonic condition, but if those things like go in the outside, that's the challenge. And here I am showing another experiment, like we did it earlier in UC San Diego. We took like 10 different microalgae species, and we grow and we make like the different kinds of consortia, like 10 different consortia for 2 species richness. We make like 10 different consortia of 5 different species richness, and we also combine all together. And we grew algae that is also your stationary phase. At that time, we introduced like a Daphnia in the system. And we have seen like the Daphnia cannot survive too much like higher species richness. It might be due to like they're not finding the food what they want. So that's why like they are going dying. So this polyculture that can you also help you to protect from your algal culture. So that is also another advantage of growing polyculture in the outside. So from these things, polyculture experiment, I would conclude like, yeah, of course they can increase the productivity, but think about, I am again telling you, what is their complementary trait, like what they're liking, like the light, temperature, what they like, the difference in that. So you have to think about it. And also like this polyculture, they can give you the crop protection, the biofuel stability. But if you look the industry, what they're doing, they're growing the super species like all the industry. They're finding a strain which can give you much more productivity. They're going in this like in the field, not like the polyculture. They're trying to get like a single species culture. But I'd say like it's not bad idea because like we have seen our green revolution just by growing single species of a like crop, like just like a paddy field here. You can see where like in green, of course, like green revolution gave us the food for us. But at the same time, actually we have lot of diversity because for this intensive farming we had to use like a lot of fertilizer, lot of insecticides, so we lost. But I think that for biofuel production we should not repeat the same thing. So we could think about the sustainable way, like how could we make the biomass and where the polyculture have possibility. And you think like those things like always comes like when you're growing in the field. So we have been working in TERI like with some rotifers. That is also a big challenge in the biofuel pond. And when you introduce like after 10 days, so when they're growing, and you can see like how their biomass are, like how the biomass, like where how the population crashed after a few days. So how do you control those things like the polyculture? At the same way, there is other possibility we could think about, and we have been doing some experiment. Like yeah, like there is some top grazers if we could introduce in the pond if they don't eat. So and if we could make like a kinds of a stable community, just like our natural system in the industrial system. So there is a possibility. So we have been working on it. So let's see like what is happening in future. But I was telling about the problem always comes in the field, like not in the axonic condition. And there was earlier there was a question like yeah, what is the challenge? Yeah, I was telling these things like. So we need to think about what is happening in the field, like in the. So we are doing a project like the field-centric process innovation for the biofuel and byproducts industry with the different partners. Two industrial partners are there: the Nanotech Hawaii and Qualitative in Imperial, Texas. And with some industrial partners are there, national lab partners are there, like the University of California, San Diego is there, New Mexico State University there, and TERI is here also. So we are growing the same species in the 5 different locations. And we are trying to mimicking the same way, just like in the right hand side, and Dr. Ramrit showed in the last videography how we are growing in the field. And we are trying to maintain like the same water depth in all the areas, and we try to find like what are the challenges we are facing, and at the same time how do you deal with those challenges like the biological, and what are the difference like if you grow in your multiple species, what are the variability is coming, and how do you explain those things. And at the same time our target, like if you grow for multiple years like the same thing, you are not like cleaning those tanks. We have started in last month, and it will be growing for like few years, and we'll want to see like what will be happening at the end, like how they will be evolved. Because our industrial partners, Nanotech, they are telling their experience, like they grow algae for your like cyanobacteria, but they're telling like, yeah, if you grow the algae in the outside for multiple years or few months even, when they're taking back in the laboratory, they are behaving differently. So what kinds of evolution that is going on? So we want to try to track those things, and at the same time, if you get like from the different climatic condition, you will get like the different strains. So in future, if you could use like mix those strains, so it is possible to make a robust community, and will get some stable biofuel production. But yeah, so I'm coming to the questions like always asking like this feasibility of the algal biofuels. Of course, like the cost people are talking, but you have to think two things before cost. Like one thing that is important, that is the energy return on energy investment. So how much energy you are getting by energy investment. So you can see the data from your Sapphire Energy Pond pilot scale facility, Las Cruces, USA. So here you can see like the biodiesel produced from your algae, that is above on, and also like their full scale facility when they're using. So yes, that is actually varying like your 1 to 3. And the biofuel is producing like from the corn, like the corn ethanol I'm just talking about, it's just above 1 or near 1, but people are using those things. But you can see imagine the achievement here, just only serious research of 15 years only, like 2015 to 2020, I would say, like just in 15 years, just like this energy return on investment. So that is a huge, I would say. So that is actually give us hope, like to think like here is the possibility algae can give us like more energy than others. Like here is the possibility, I would say. But people always talk about the cost first before these things, but you can see like how it would be. Like in the right hand side, here this cost actually, algal biofuel cost, it is estimated based on the algal productivity. If you look on this blue green line here, that is only selling only the biofuel, if you use these things, because like the dotted line here, it's showing like the around $3 per gallon your oil price. So we cannot reach, because like with our existing technology. So because in this oil price at that time, like today oil price, how much like this, I had seen in the morning, it's like $60, $50 in international market. So we cannot compete it like with only using the biofuel currently with our existing technology. I'm just focusing these things. But if our technology is like enhanced, just like if you could find like some technology for your harvesting, like the low cost technology, yeah, we could achieve it very easily. But at the same time, but if you extract like the red line, if you fractionate like the different products, so at that time we could reach like those things. So like that's why people are thinking for your other products like from the algae at the same time with your biofuel. And industry, like those industry, like I'm still like in 2010, like they have started, like they have invested their money for the biofuel, but they have shipped or switch their idea for your other product. But otherwise, like some, except for your synthetic genomics, other companies not only focusing your only for the biofuel. So they're using like the other products at the same time. And in India, I would tell, like of course, India have a huge prospect of the algal biofuel or other products because they have the favorable climate. So like this publication, like one of our actually collaborators in the US, so they published this paper in PNS. So you can see like the red means your like high productivity of the algae, and your the blue means your low. So if you look at these things here in India, that is actually huge potential because of their climate. And at the same time, India have a lot of wastewater that is untreated. So this water would be a source for growing algae. And at the same time, the water, actually, India have a lot of coastal line, more than 7,500 kilometers. So we could use the coast, like people you can see like living in your miserable conditions in the coastal region. So there is a huge prospect. So we could use those things. And before going in my talks, like I'm just telling like some of the questions like from the participants, like one question from the Pauline Sarkar from IIT Guwahati, like if you imagine grow algae in algal pond for large scale production and use natural sunlight, the algae at the bottom will get less light, of course it will. Is it possible to use artificial light for the lower part and use sunlight for the upper part of the pond? Yeah, we could did it because but first of all, I'm telling you, Professor Chisti actually published a paper in the real scale, not in you're talking about your natural pond, but in your raceway pond, even like when they're mixing properly at that time also 90% of the time a single cells are living in light limited conditions. So yeah, it is possible, but depth is a matter, and yeah, there is a need of survey in which kinds of ponds are available in India, like the low depth pond, and we have to think like the light you are talking about. So, and at the same time, I am telling you the low few species of are there they like the shade at that time, just like the diatoms. They're like the low light intensity. If you could grow at the same time your highlight intensity, so there will be a chance of growing multiple species. So that will be on chance. So yeah, so that is is it possible to village up India? Yeah, so it is possible, but we have to survey on it. And also the biofuel industry, they're asking, yeah, I was just talking about prospect of biofuel industry. Yeah, so most of them are going for the parallel products. So, and also the algal oil, how it is separated from algae, Dr. Romit told about it, and economics, yeah, I'm telling you this biofuel stuff, currently people are much more focusing on the not extracting the lipid and making for the biodiesel, and but mostly they are trying to use the total hydrocarbon of the algae, and they're trying to convert in your hydrothermal liquefaction and converting into biooils, because at that time the you can use drying is also a major factor, so that is also another prospect. And the last thing I answered it already, and there is a video clips. So one is high throughput screening. So just before going to this video clip, I'm just telling you it's how do you measure your algae bacterial interaction, measure the algal growth. So we have seen this chlorophyll fluorescence that we could measure it it's independent of your bacterial population. We have tested in laboratory, and using this your 24 well plate, and if you combine with your adjustable spacer multi-channel pipette, so it would be much more easier. It is much more faster. So you can test thousands of combinations that's in a very short time. So not only for the algae bacterial interaction, the people are working on your different nutrient stress. So those kinds of study that you could use it here very quickly. So this process. So that's why I want to show that's a very short video, and our student Gurpreet, she actually made all those things. So let's see. Thank you.
So thank you, Dr. Shawan. It was a very nice presentation and interesting lecture. We have some questions from the participants. So the first question is from Professor Dr. Rangarao Ambati. And the question is, what type of predators did you observe in ponds or PBRs?
What type of kills?
What type of predators did you...
Predators, okay. Yeah, so actually mostly like when I grew like in the outside, like that is the rotifers, that is the things. And we have seen like a lot of times like the amoeba attack, like so paramecium kinds of thing, like you frequently find those two things.
Okay. The next question is from Dipali Shukla, and she is asking, are we taking any specific species of algae?
Yeah, like for the specific species, like so that's a like it's not clear, but I'm just telling, yeah, like you, it depends upon your target. Like if you're thinking for your particular products, like so you have to think about it like a particular species. Like if you like think for your Fico, you have to think for the cyanobacter species. But for the biofuel, when you are thinking for making consortia, like we don't think like all are fast growers, but we are thinking for the species they are having your complementary trait. Like some species is liking on things like liking on your highlight intensity, other is liking your low light intensity, or like your different spectrum. Like if you grow like cyanobacterial species with your green algae, what will happen, like they are having your different spectrum of light they can absorb, so there is a chance of a higher productivity. So in that way we will select the species.
Third question is from Dr. Ram Kumar. Can we supply molasses spent wash as substrate for the growth of algae, and will the product like spirulina can be claimed as organic product?
Yeah, like this molasses, like the organic, like you're talking about this mixotropy condition, like in the field. So, yeah, like in the open pond, like you could, there is a challenge like for the other bacterial species that can come in, and uh, it's not like this organic product means like your, what do you want to say? It's not clear to me also again. So yeah, but for the mixotropical, yeah, always that gives you much more productivity. You could use it.
Okay, so with this, I would like to thank you very much for sharing a broad spectrum on algae potential and the future prospects. It was a very interesting lecture. Thank you so much.
Thank you.
So now moving towards our next lecture by Professor Rangarao Ambati. Dr. Ambati have been working as a senior scientist and associate professor in Department of Biotechnology at Vignan Foundation of Science, Andhra Pradesh. Recently, he is identified as top 2% world scientist reported by Stanford University, USA. Dr. Rao worked as a lead scientist in Algal Technologies, Carrot Lab Private Limited, postdoctoral research associate in Laboratory of Algal Research, and visiting assistant professor in Food Science and Technology program. He published more than 50 peer-reviewed publications, 50 international national conference symposia, invited talks, and 14 chapters in book. He was selected Junior Scientist for Air Award 2015, a National Environmental Science Academy Award, New Delhi, and also he's the Associate Fellow of Andhra Pradesh Academy of Science 2019, Government of Andhra Pradesh, India. So now we welcome Dr. Rao to deliver his lecture.
Okay, thank you very much. Am I audible?
Yes, you are audible.
So I want to share my video, but it is not at all, it's not taking.
IT team, please give the access to Professor Rangarao.
Yes. Now it's okay. Thank you. Is it visible, my slides?
Yes.
Yes. So can I get the access, because the next slide I cannot able to move. Hello.
You can use your keyboard to move on these slides.
Yes, yes, I'm doing, but
So just give us a minute and we'll
yes, yes, sure, sure.
get back to you.
Yes, yes.
Dr. Rangara, our side,
Oh yeah, sure, sure, madam.
you can email to me or
Yeah. Okay. Yes,
so
just
Dr.
now
Amadi.
just yes, Dr. Ruchi.
Okay, yeah. I'm waiting for that. I've
Oh,
not
okay.
received it yet.
I sent you Ruchi Agarwal, doctor.
Uh, Ruchi call
Yes.
@TERI.rest.in.
Yes, madam. I sent you that. Ruchi.agarwal at the rate of TERI.resin.
Okay. So we'll wait for your presentation as soon as we receive so we can play it.
Yeah, well, otherwise, I will try one more time, please.
Yeah.
Can I?
Let's mail if you have,
Yes, madam. I have that one.
because I have not received. Maybe there's some error in the
Yeah,
ID.
okay. Yes, I will forward. I forwarded, madam.
So Dr. Mbati, we have received your presentation and we will be playing in next 1 or 2 minutes. It is
Oh,
there.
okay. Thank you very much.
Can you see the presentation now?
Yeah. Yes, I can see.
Okay.
Yes. Okay. Sorry for some technical problem here. So, can I start, madam?
Yeah, please.
Yeah. Okay. So first of all, I would like to thank to Dr. Alok, Program Director, TERI, and then Dr. Amritpreet Kaur and Dr. Ruchi for sending me the invitation for this workshop. So today I am going to talk about encapsulation of microalgae and its metabolites for food, pharmaceutical, and cosmetic applications. So next slide, please. So previously, I worked on various international organizations. I worked in Arizona State University. We focused more on microalgal cultivation system in photobioreactors and racewater ponds. Just now, one of the speaker talk about the Dr. Shawn Mundel. So how the predators dynamics in mass algal cultivation system during the cultivation system that we worked there in Arizona State University in different systems, racewater ponds and photobioreactors. Coming to University of Malaya, I compare the productivity using various cultivation systems, various photobioreactors, and including the racewater ponds. And coming to CFTRI, I did my PhD from CFTRI, and we worked on more aspects, metabolites from microalgae and hydrocarbon production from the microalgae. We have worked we focused more on this aspect. And I worked in China. China also we worked on the biomass production from microalgae. So next slide, please. So majorly I worked on various microalgal species and then cultivation aspects and then applications oriented how these metabolites can be used in various applications, food applications, and also we worked on bioremediation applications when I was working in Malaysia using wastewater treatment. We have done that also. And then enhanced the productivity in terms of biomass, lipids, and then hydrocarbon. And coming to downstream processing and then harvesting. And then we also evaluated the characterization aspects, various metabolites from algae. And then we also checked the in vitro and in vivo metabolites, in vivo model systems, how these metabolites can be used, especially, astaxanthin, from the Hematococcus fluvialis. And then safety assessment also we studied this algal biomass. So these are the contribution I have done so far in the algal biotechnology. Next slide, please. So as we know that algae is a photosynthetic microorganism and then accumulate various metabolites, secondary metabolites, and the primary metabolites, and the secondary metabolites, especially we are focusing on secondary metabolites. So these secondary metabolites, it will be vary the culture conditions, as just morning spoke about Dr. Preeti. She talked about the culture conditions and then various pH and then temperature and how this will be vary metabolite production or biomass or lipids. And this is one of the important one. The metabolite production vary upon the culture and then environmental aspects, environmental conditions. And then, as we know, we are seeing that these metabolites can be used in various applications, as I mentioned, food, feed, nutraceutical aspects. And algae produces various bioactive compounds, proteins, polysaccharides, polyunsaturated fatty acids, and pigments and phenolic compounds. So lipids, these are people are using for various purposes. Next slide, please. So why we want to focus on metabolites? Why we want to focus on metabolites? What is the importance of these metabolites? But if you see the consumers, currently consumers are looking for the natural ingredients, natural metabolites. So especially from the natural sources, people are looking for the metabolites, and then they want to use in various applications. As you know that this algae produces metabolites, and they are safe and then environmental sustainable, and they are not toxic. and the comparative synthetic ones for various nutraceutical applications. And these metabolites, significantly worked on various biological aspects, antioxidant, anti-cancer, anti-inflammatory, antimicrobial, and immunomodulatory aspects. The people, these are some of the few studies, focused on this one. And in addition to that, people worked in various aspects in terms of biological activities, in vivo, in vitro studies. Next slide, please. So these are the algal products available in the market. As yesterday spoke about Dr. Sunil Pabbi, and then Professor he spoke about producing spirulina biomass and phycocyanin. People are using in various applications, and it is available in the market. And similarly, astaxanthin people are using for various applications, and chlorella biomass they are using, and omega oil from microalgae they are using. So these are some of the products available in the market. Next slide, please. And these are the commercial species. The last yesterday and today people are talking about this one. So commercial algal species, spirulina, and then chlorella, and dunalella, and then hematococcus. So these are the potential species people are working for commercial applications, commercial aspects. Next slide, please. And we are talking about the metabolites. So what are the metabolites? If you see the metabolites, spirulina produces phycocyanin. We can extract from the phycocyanin from spirulina, and astaxanthin from hematococcus, and the beta carotene from the dunalella, and the PFAS and phycoerythrin from other species. And then we can also get omega-3 from the spirulina, and then beta 1,3 glucan we can get from the chlorella species, and fucoxanthin, and also EPA. So these are some of the metabolites people are using biological activities, anti-tumor, antioxidant, and the antibacterial, and the hepatoprotective, and the anti-cancer, and then high protection, and then immunomodulatory aspects, and then improving insulin resistance. And these are some of the biological activities people are studying, reported about this metabolites from the microalgae. Next slide, please. So today my talk is, what is encapsulation? We are going to talk about the encapsulation. What is encapsulation? Because here we need to remember there are two. One is active ingredient, what are the metabolites we are going to use, and the coating material is very, very important. So here we need to remember these two. So it is a process, encapsulation is a process, but we are going to use the active ingredient, which the metabolites we are going to use. But why we want to use the coating? We need to protect our metabolites. While you are using the protector, we need to remember this one: control, protect, and the deliver. These are the three aspects it is working on this one encapsulation process. But the metabolite we are going to use, that should be a safe, environmental safe, and we need to deliver for biological applications. And when you prepare these nanoparticles, the range would be 1 to 100 nanometers. When you use micro encapsulation process, that range should be 100 nanometer to 1000 micrometer. This should be the range of the dimensions of this nanomaterials. Can I have next slide, please? So what is the advantage of this encapsulation process? So we need to remember this one: maintain biological and functional and physiological properties of the active ingredient. What are the metabolites we are going to use? We need to look into what is the biological and then functional aspect and the physiological properties. And we need to protect environment, processing conditions. Okay, that is very, very important. We need to protect the environmental and then conditions, environmental aspect, and the processing condition. During the process, it should be lost. It should not affect any light or other storage conditions. And then increase stability. We need to increase the stability and then shelf life of the particular ingredient. And the solubility is very, very important when you use for some biological activities. And then mask and pleasant flavors, and enhance the bioavailability. When you use this one, we need to see the bioavailability. It should be very, very important. When you use biological activities, we need to increase the bioavailability. How this it will flow through the blood, and it will do the organs, then how it affects on the biological activity. And then control the target release of the core materials as a response to the target, where we want to target, whether we are looking for the anti-cancer, whether we are looking for the some other anti-immunomodulatory effect, or we are going to use anti-skin cancer oriented. So we need to see where we need to looking into the biological activity. That is whether it is delivering properly or not. So that is also important. The process, that is the reason we are going to use this encapsulation process. Next slide, please. So when you select the coating material, we need to look into the structural, chemical structure and molecular weight, and the polarity. We need to look into the polarity, whether it is a lipid soluble and water soluble, whether it is a lipid soluble and water soluble. We look into the how it works, how it is a solubility, whether it is increasing or decreasing, or the delivery, how it happened. So we need to look this one, and the efficiency and the permeability and degradability, whether it is a degrading the particular this one, and release efficiency. We need to look into this when you select the coating material. When you select the coating material in the encapsulation process, we need to look these aspects. Next slide, please. And then encapsulation process people are using in diverse areas in scientists and the world. Food aspects people are using, pharmaceutical industry using, cosmetic industry. Now the cosmetic is looking, most of the people are using cosmetic products today. So this encapsulation process people are using, and then chemical industries and the agriculture aspect. So the encapsulation technology using various industrial aspects, industrial applications. Next slide, please. And then when we talk about this encapsulation process, what is the structure and composition of encapsulation process? If you look into here, the image, the figure A, B, C, D, and E. A is, you can see here, figure A. This is a monocore. This is, you can see the shell round, okay, a single cell core, okay, around the material, the active ingredient, okay, that is a outer shell around the single core, okay. So coming to figure B, if you look into the multi-shell, there is a black shell and then red shell and a blue shell, but the in between the active ingredient, that is a multi-cellular one. Coming to multi-core, surrounded by the single cell. So you can see 1, 2, 3, there are 4 or 5 cells are there, but it is around, you can see the coating materials. Coming to matrix. Matrix is the active agent distributed with a coating, okay, that is mixed together, that is a matrix, okay, the active ingredient and the coating material, that is a matrix. And the coming to irregularly, we can see this is a shape of the particles. So this is structure, and then composition is very important when you take this process, encapsulation process. Next slide, please. And what are the active ingredients we are going to use in this encapsulation process? We are going to talk about the metabolites, carotenoids, that may be vitamins, minerals, and drugs, nutraceuticals, antimicrobial, antioxidant, flavors, enzymes, and essential oils, and the coloring agents, beta carotene, astaxanthin, lutein, some other. You can see here, right side, there are encapsulated essential oil in biopolymers, and then encapsulated vitamins, and then encapsulated minerals. So, and then some of you can see samples produced using different encapsulation technologies. So these are some of the images which I got from web sources. And coming to next slide, please. And coating materials. When you select the coating materials, what type of materials we need to select? People are using natural polymers and then synthetic polymers and bio-based substances. When you take these natural polymers or synthetic polymers, we need to look into the solubility and the stability and the releasing properties and the safety aspects, safety regulations and safety aspects. And then biocompatibility, how the biocompatibility and the biodegradability. We look into this one when you use these coating materials for the encapsulation process. Next slide, please. Coming to natural materials, people use natural polymers, okay, carbohydrates, starch, maltodextrin, pectin, cellulose, cyclodextrin, and inulin, and proteins, gelatin, whey protein, and casein, bovine and wax and proteins, and gums, different type of gums people are using, and polysaccharide, chitosan, and then alginate. These are some of the natural materials people are using as a coating materials in the encapsulation process. Okay, next slide, please. Coming to synthetic one, these are some of the synthetic one. There are biodegradable and non-biodegradable people are using. The biodegradable materials, aliphatic polyesters, and then coming to non-biodegradable cellulose derivatives. Okay, aliphatic polyesters, polycaprolactone, polylactic acid, and then polygogalic acid, and polyanhydrates, and polyamides, and polyurethanes. People use this one, biodegradable materials for the coating materials. And then coming to non-biodegradable materials, cellulose derivatives, carboxymethyl cellulose, ethyl cellulose, and then polyethylene glycol, polyvinyl glycol, and poly-N-vinyl pyridine. So these are some of the biodegradable and non-biodegradable material people use for this encapsulation process, but these are the synthetic sources. Next slide, please. And this is one of the example that chitosan people use it for this one encapsulation process. You can see here, the first one is chitosan, and then active ingredient, the red color, the dots you can see here in the image. And then encapsulation process, they mix it together. You can see here encapsulation process during the process. Then this is encapsulation ingredient. And then this you can see the red color one, and then blue color, and then middle is the yellow. This is a shell. This is a encapsulation cell, and then core materials. And then coming to core release, how the drug it is releasing, you can see here there are red dots is coming out from these materials. So, and this can be used in food, biomedical, and then agriculture, textile, and pharmaceutical, tissue engineering applications. People use this chitosan. Next slide, please. And encapsulation technologies. What are these technologies? There are several technologies people are using for this encapsulation technologies. Spray drying process and spray cooling, fluidized bed coacervation, solvent evaporation, liposomes, supercritical fluid technology, interfacial polymerization, and then nano precipitation, emulsification, and then electrospray system and extrusion. These are some of the nanotech. This is encapsulation technology people are using for the encapsulation process. And then when you select encapsulation technology, we look into the cost effective and then easy to scale up. We need to see the cost effective manner, and then easy to scale up, and then safety. We look into the safety aspect. So these are should be considered when you select the encapsulation technology. Next slide, please. And there are two approaches during this process, encapsulation process. So there are two, one is bottom-up approach and the other one is top-down approach. So bottom-up approach, what the bottom-up approach will do, the large structures are built with atom by atom, okay, or molecule by molecule, mediated by different interactions. The interactions between, you know, van der Waals or ionic and hydrophobic interactions happen when during this process, bottom-up approach. But approaches includes chemical synthesis. People use chemical synthesis process and cell assemble and positionated assemble and molecules. They will use this bottom-up approach. And also nano precipitation. And this nano encapsulation process, this is a technique. In this technique, they will use this bottom-up approach. And coacervation and supercritical fluid extraction also, they will use this bottom-up approach. And coming to top-down approach. In this approach, what is important? The core and coating material, which requires precise tools focusing the size is important, and the reduction and shaping of the structure. These are the very, very important. In this process, we can see this one top-down approach. But coming to between these two techniques, bottom-up and top-down approach, the bottom-up approach is one of the more advanced than the top-down approach, and the people are using many of them this one. And but this approach allows the control over the properties of the particle size and the morphology and the physical state of the and the less energy consumption and contaminants. So these are the aspects that is advantage. Bottom-up approach is better than the top-down approach. Next slide, please. And then micro encapsulation. So as we know that yesterday and today we are talking about there are microalgae produce valuable products and they are economically viable and we can use for various purposes. And then however, people are used in various food aspects. People use cookies, bread, pasta, and dairy foods. They use this microalgal biomass, fortification, and then incorporation. They use nutraceutical aspects. That is but here the problem is incorporation is one of the important one. That is one of the major challenge when you do this microalgae encapsulation process. The effect of the biomolecule when we are using the health food applications, look into its function of the intake during the food processing and the storage, characteristics, accessibility of the original food, and the bioavailability of the particular metabolites or active ingredients. We look into this one when you use this micro encapsulation system. Next slide, please. Due to the inherent of the most bioactive compounds in algae, the efficiency of this process is compromised. Therefore, the incorporation of this encapsulation process is promising strategies to deliver microalgae health benefits using this technology. And however, some of the species are used, Hematococcus and Spirulina, Dunaliella and Chlorella and Piodactylum trichonium. These also people are used and reported in terms of the extracts and the biomass and then isolated compounds from these algal species. in terms of the pigments, astaxanthin, and the beta carotene and phycocyanin and phocoxanthin. So these are used for this encapsulation process. Next slide, please. And encapsulation process of food applications. So we are going to talk about this one. Can you please, next slide, please. So if you see the astaxanthin, here look into the astaxanthin. Now the trend is people are looking into the astaxanthin because astaxanthin has more applications in terms of biological aspects. So this is the techniques people encapsulation process. They use hydrogel nanoparticles and polymeric micelles and the polysaccharide beads and cyclodextrin and then water and then oil emissions and then some of the polysaccharide liposomes and polymeric nanomaterial. These are the people use it for the various process for this encapsulation process using astaxanthin. Next slide, please. So this is actually people use food applications of Haematococcus culture or astaxanthin. They extracted from the astaxanthin. They have used this using encapsulation process in various food applications. But if you see the look into this table, coating materials, there are different coating materials, chitosan, polymer, hydroxy, irritated co-hydroxy literal and chitosan and then chitosan, carrageenan, calcium, alginate. So if you look into this table, there are different materials, different polymers they use with this encapsulation process. And then look into the second column, encapsulation technology. They use a different technology. And then you supercritical fluid and extrusion and polymeric nanospares and liposomes. So these are the reported this one encapsulation technology using this astaxanthin. And what they observe, the major one is this encapsulation process is achieved when using higher biomass and increase the stability under thermal conditions and the storage conditions. And then when you use this chitosan, okay, beads provided higher protection to astaxanthin. And then alginate beads when compared to the alginate beads. So overall this one, so when you use this encapsulation technology, especially this metabolites from algae, so improved the stability and then enhanced bioavailability and then stability under various culture conditions or various temperatures. When you use this various temperature storage stability condition, improves the this efficiency of this metabolites. Can I have an next slide, please? And then coming to this optical microscopic image, excuse me, can you? Oh, can I have before slide, please? Before. Can I have before slide, please? Next. Okay, next slide, please. Okay, this is we talk about the Haematococcus. Coming to Spirulina, Orthospira species. Can you please go for next slide, please? Yeah. Okay, this one you can see this is Orthospira species. This is Spirulina. So people worked on similarly for as we have seen the Haematococcus. So people used various coating materials and then various encapsulation technologies for doing the encapsulation process, the spirulina and then phycocyanin from the spirulina. So during this process, thermal stability improved for phycocyanin and showed higher antioxidant activity, encapsulation efficiency improved, and then maintained the, they use yogurt. We are taking yogurt. That yogurt nutritional profile, it is not changed. The improve the antioxidant activity through the storage time and showed a higher antioxidant activity when they used nano dispersions. And then micro encapsulation protected the biomass antioxidant potential in pasta and in various foods. And then formulations also they use this encapsulation technology and increases the stability conditions. Can you go for next slide, please? And this is also other algal species, that is fucoxanthin from the Phaeodactylum trichonium. They also prepared the nanoparticles and they exhibited superior bioavailability in vivo models, and the encapsulation process showed efficiency is very high when compared to the other one, and then promoted stability, improved the beta carotene in Dunaliella species. Beta carotene extracted, they used encapsulation process, and then Chlorella also, they prepared this proper encapsulation technology, and then they prepared, and they also showed that inhibitory effect on the human cells, human cell lines, and the preserved against enzyme degradation, increased the bioavailability. And similarly, you can see different species, they extracted compounds, and then they checked this encapsulation process and enhanced the activity, antioxidant bioavailability using this process. Can you go for next slide, please? And astaxanthin, we are talking about more Haematococcus, but whereas you can see astaxanthin extracted from the shrimp and extracted from some of the bacteria, and they processed this encapsulation process, encapsulation technology, and they used various polymers materials. You can look into the column 2. There are various polymers materials they used here, coating materials, and then encapsulation methods. You can look into the last column. The efficiency, it is increased. It will be vary, depends on the polymers. It will be very depends on the polymers and then encapsulation technology. So the efficiency, it is very high when they use solvent displacement, okay, polyethylene oxide, and then you can see supercritical fluid extraction. Then they use starch as a polymer. And look into the here some of the alginate, when they use the alginate. So these are the efficiency of this encapsulation technology when you use different polymer materials and different encapsulation process. Next slide, please. And this is one of the emulsion we have taken. I have taken this one from the reported by author. So encapsulation of this astaxanthin, you can see figure 1, and the figure 2 is scanning electron microscope, and then figure 3 is TEM, transmission electron microscope. And whereas you can see here figure D, here it is showing that dimensions. You can see the dimensions of nanoparticle, the intensity, okay, x-axis and y-axis. X-axis is the dimension particle size, and then y-axis percentage of intensity. So you can see the varies the size of the nanoparticles. Next slide, please. And coming to encapsulation process from the shrimp, actually there some reported that they extracted the astaxanthin from the shrimp, and then they prepared the micro encapsulation, and then you can see here the encapsulation stability is increased del 2 based on the time interval 0, 10, 20, 30, and 40, and 50. Look into the figure here. The process, the stability is increased when the when they prepare these nanoparticles with the polymer materials. Next slide, please. And this is the spirulina. From spirulina, people use they prepared the food products, and then they prepared cookies, and they also used the encapsulation technology here, and then that encapsulation process. But you can see here biomass and the free extract and then encapsulated extract. You can see the comparison here, and some of the images we have taken from the web sources, and then the encapsulation process efficiency increased. Next slide, please. Coming to pharmaceutical, as people reported the food applications, this is some of the pharmaceutical applications. People use algae for various applications. Can you go for next slide? Similarly, as I mentioned that this spirulina, people use it for the pharmaceutical applications, different coating materials and different nano-encapsulation process. And the major, they observed that some of the biological activity that C-phycosidine loaded chitosan with excellent mucoadhesive properties of colonic drug delivery, and the higher antimicrobial activity, slower release of profile when compared to the like a free form. So these are the some of the reported this pharmaceutical applications from the microalgae. Can you go for next slide? And this is from Haematococcus. They did work on Haematococcus, and they use it for the some of the biological activities. They prepared here astaxanthin extracted, okay, loaded cellulose acetate nanofibers, and they see that against oxidative stress of the Cl gains with significant values, the stability of the nanofibers was significantly higher compared to the free extract under exhalated conditions. And the encapsulated astaxanthin inhibited cells, cell lines, but it had a relatively small or no impact on control hepatocytes. And astaxanthin reduced the lipid damage when different lipid peroxidase promoters were added simultaneously to the liposome. And other showed this antioxidant, this astaxanthin hepatoprotective and anti-inflammatory activity by astaxanthin loaded liposomes nanoparticles, and then improved stability and then bioavailability, and improved when compared to the free, and observed that bioavailability increased when compared to the free molecules, free form of this astaxanthin. And also encapsulated astaxanthin able to control the or induce the reactive oxygen generation apoptosis through the apoptotic signaling pathways. This is some of the studies that reported Emetococcus, astaxanthin from Emetococcus for pharmaceutical applications using nanocapsule system. Next slide, please. So coming to cosmetic application, next slide, please. So cosmetic nowadays, we are looking for the cosmetic applications from the metabolites we are going to use. So this is very, very important. Most of the consumers are looking for the natural sources, natural ingredients, and then the metabolites we are extracting from algae. We are going to use the skincare products, skin health, and the treatment of the dermatology issues and the hyperpigmentation, dehydration, photooxidation, photoaging, and skin cancer. We also worked on skin cancer, anti-skin cancer of astaxanthin ester from Emetococcus. We also reported and published papers. And astaxanthin and phycosine also reported in cosmetic applications. And then that encapsulation process, especially the liposomes, was well performed in vitro in vivo models. Can you go for next slide, please? These are some of the report that cosmetic applications of algae for various algae enhanced astaxanthin to protect antioxidant in vitro in vivo models, and against UV-induced UV damaged skin damage, and then inhibited collagen degradation, melanin production, and then protected cells against oxidative stress, and then showed higher antioxidant compared to the pure astaxanthin after exposed to UVBA radiation, and restored mitochondrial membrane potential. So, and then spirulina also extracted nanofibers patch found to be non-toxic in human cell-based system, and then promoted the release of the most of the extract onto the skin within the 30 minutes. And then phycosine also protected the stress human keratositis when compared to the free form, and control in inflammation and stimulates and keratositis proliferation. So these are some of the studies that they used cosmetic applications for microalgal metabolites using the encapsulation process. Next slide, please. So this is one of the study we conducted that this stability of astaxanthin we dissolve in oil. We studied some of the stability studies. We found that this astaxanthins more oils, and then especially if it's the rice bran and then gingerly, palm oil, they retain 80 to 90% of the astaxanthin when heated 72 degrees and 8 hours. So what we did is we did stability at different temperatures, 70, 90, 120, 150, and we found that astaxanthin is stable even higher temperatures. And then especially if you look into the palm oil, is effective in retaining 90% of the astaxanthin at 90 degrees for 8 hours without any change in ester form. You can look into the here red color. This is a TLC. We found that there is a very big pink pigment, and then 90% of the carotenoids is retained. And whereas if you see the water form, we also dissolve in the water, but what we observe that in compared to the oils, the water, we lost the carotenoid content in aqueous form when compared to the oils. Next slide, please. This is the peroxide value. What we found that when we see the oils, when we talk about oils, usually we measure the peroxide value. It is very, very important. When they actually happen oxidation, then the peroxides form in the oil. So what we observe here, when we tested this one, stability studies, so compared to the control group, initial this one, that peroxide values, okay, the peroxide values are reduced where we use this astaxanthin extract in edible oils. That was I want to say here. Next slide, please. This is HPLC. We have done that carotenoids in rich edible oils. The first one is control group, and whereas you can see here B and C is the astaxanthin esters are stable when you use the even higher temperatures. Next slide, please. And further, what we did is how the bioactivity of this even we store the oils longer time. room temperature, and we tested various temperatures. Then further we thought of that to check the biological activity, antioxidant activity in vitro models. So what we observed that this astaxanthin showed this showed antioxidant activity in vitro studies in various edible oils. You can see here there are several oils, and there are two methods we used, antioxidant methods, DPPH method and then hydroxy radical scavenging activity. Next slide, please. So the conclusion, so we are this encapsulation process, so incorporated with either algal biomass, algal metabolites, isolated compound. So when we applied food or pharmaceutical or cosmetic and nutraceutical applications, the presence of coating material is promoted a stable enhancement under different storage conditions, suitable bioabsorption, skin permeation, and improved physiological properties, control the compounds release in target site, and proven that effectiveness, stability, improved the bioavailability in various studies. And then astaxanthin from Hematococcus, phycocyanin from spirulina, other algae were well studied in various applications of this encapsulation process using this encapsulation system. Next slide, please. So overall, these bioactive compounds showed potential antioxidant activity and positive effect in terms of several disorders. And astaxanthin and phycocyanin consider the health promoting substance in diverse areas. And these encapsulated bioactive compounds showed a higher therapeutic value in various studies. And astaxanthin from Hematococcus, beta-carotene from Dunaliella, for commercial encapsulated microproducts are available in the market. Therefore, the current this encapsulation process, microalgal derived compounds or encapsulation process advantage is promising. This is a technique promising strategy to develop innovative methods or healthcare products or various applications using this encapsulation technology. Next slide, please. So these are some of the books we published. This handbook of algal technologies using food, health, nutraceutical applications from CRC Press, and volume 2 also we published pycoremnation, biofuels, and global biomass production. And recently, this global prospect of astaxanthin, this book we edited from elsewhere through elsewhere association publishers. So it may be published in the next month, April 2020. This one schedule this book going to publish. And next slide, please. Myself and Dr. J. Ravishankar, we both are editor for this book. And these are some of the publications, my work. Next slide, please. Next slide, please. These are some of the publications from my publications. Next slide, please. So recently, I identified, reported by the Stanford world top 2% scientist by Stanford University. This we got from the University of Malaya, because when I was working there, we published papers and highly cited that paper. And maybe that may be the reason my name will be there in this one, world top 2% scientist by Stanford. Next slide, please. These are the research groups. I worked with Arizona State University, Professor Milton Somerfield and Dr. Q Hu. These are the two co-directors. I worked as a postdoc student under their supervision. They are co-directors. And coming to University of Malaya, Professor Pang Simoyi, she is my supervisor, and I worked with her there, University of Malaya. And third slide, when I was working in CFTRI Mysore, and then I working in China, when I was working in I have taken photo with the group of people. I have association with them. Next slide, please. So finally, my acknowledgements. Thanks to Dr. Alok, Program Director, Dr. Amritpreet Kaur, Dr. Ruchi Agarwal, TERI, Deakin Biotechnology Center for inviting me, management of Vignan's Foundation for Science, Technology and Research University, and my mentors, especially I never forget this, my mentors. Because of them today I am here. Professor Dr. J. Ravishankar, Vice President, Dayanand Sagar Institution, Bangalore. He was a former head chief scientist, CSIR, CFTRI Mysore. And my another co-supervisor, Dr. R. Sharda, former head chief scientist, CSIR, CFTRI India. And also, I associated with Dr. Uday Shankar, Dr. Shailaja Dharmesh, and Dr. V. Bhaskaran. During the animal studies, I associated with them. So we did some of the work and we published. And then coming to my foreign supervisor, Professor Milton Somerville, I think unfortunately is no more. So Professor Kyuho, Arizona State University, USA, and Professor Pang Simoyi, she is my supervisor when I was working University of Malaya. Next slide. Thank you all. Open for the
So we'll
questions.
now take few questions. So the first question by Mr. Gupta: how chlorella species was used for wastewater treatment, and what about ethical concerns associated with it?
Actually, we isolated this chlorella species when I was working in University of Malaya, and what we did is we have not taken any because the university has approval, that University of Malaya, but we did is we in terms of nutrients, what is the uptake of the nutrient, this using this chlorella species, we did we got this because in Malaysia there are a lot of palm oil industries. We got palm waste, and then that one palm waste contain a lot of nutrients, nitrogen and phosphorus and some other nutrients. So we checked the how to we want to see these nutrients, how to utilize this microalgae instead of using the normal media. This is how actually we did work in Malaysia.
So the another question by Ravi Kumar: can we use nanoparticle for coating? If so, how? Select the binding agent. Any alternate method is there for ultrasonication?
So ultrasonication, actually we have not done. I am very sorry about this question because I have not done this one.
Okay, so we have these two questions and now we'll move forward. So we thank you, Dr. Rao, for a very informative and enlightening presentation. So we move to our now next speaker.
So the last lecture we have invited is from Professor Munish Puri, and he is here. Munish Puri, Dr. Munish Puri is an associate professor, medical biotechnology, Flinders Health and Medical Research Institute, College of Medicine and Public Health, Flinders University, Australia. He has H-index 40. He holds a PhD in industrial biotechnology from India and did his postdoctoral training in the area of protein structures at University of Oxford, UK. He had the privilege of working as a University of New South Wales Endeavour Fellow at University of Queensland, Australia. Dr. Puri has worked as a full professor, associate professor in the Department of Biotechnology, Punjabi University, India from 2003 to 2009, where he developed the area of enzyme biotechnology before moving to Deakin University, Australia as a visiting professor and senior research fellow. He has served as the laboratory head of industrial biotechnology at the Center of Chemistry and Biotechnology, Deakin University, and has significantly contributed towards the development of bioprocessing research, where emphasis was laid upon bioprocessing of various bioactives at Flinders from 2017 to now. He is leading the area of bioprocessing and has worked with international industry partner on single cell oil production. Dr. Puri has published more than 178 scientific articles, including 131 peer-reviewed journal papers, 3 patents and 14 book chapters, mainly in high-impact biotechnology applied microbiology journals. So I would request Dr. Munish Puri to start with his presentation, please. Dr. Munish Puri, are you going to share your presentation now?
Yeah, can you hear me?
Yeah, yes.
And can you see my presentation?
Yeah, it is being shared. We can see it on our screen and you can put it in slideshow mode.
Sure.
Thank you, sir.
Okay, so can you see my first slide, all of you?
Yes.
All right. First of all, thank you, Alok, and thank you, TERI team. Amrit, Ruchi, Vikrant. It's been a pleasure talking to you for the last few days. And believe me that I was so nervous today because I was hosting a seminar and then in parallel, I had to come for the talk. My apologies that the things got clashed, but thank you for your understanding and for changing the schedule, which I really appreciate. Thank you, TD team. And thank you all the audience for going to be here for next half an hour or so. I will do my best to excite you what I'm doing for the last few years. The first slide is basically a glimpse of the area where I am. This is in south of Australia. And okay, so can you see the second slide?
Yes, we can.
Okay, all right, excellent. So I work at Flinders University, and before coming to Flinders, I was a deacon, and I was deeply involved with the Deakin Biotechnology Center, and Alok has been a pillar, and he has been guiding and advising on various things. And really, the center is Deakin TERI Nanobiotechnology Center is emerging as one of the stalwarts of leading technology in this area. And fortunately, I'm working in this area for the last 12 years. And consequent my move to Flinders University, I'm going to give you some introduction to Flinders University. Won't take much time. Yeah.
Manish, can I interrupt? Can you go to the full presentation mode? Because I can see both the of your coming slides as well on the screen. So if you go to a full presentation, would be better.
Okay. Is it on? It's on a full mode.
I can always see your next slide coming.
Okay, just a minute.
Yeah, yeah. If you go to the display setting. Yeah, I think you can click this.
Is it okay now?
No. Yeah, now it's okay.
Yeah, okay.
Perfect.
Yeah. Thank you, Alok, as always. Yeah. All right. So this is something which I thought that I will share with you. Like, it's a beautiful city, and it has been regarded as one of the best livable city. So the university where I am at the moment, this is amongst top 50 unis and #1 in South Australia. And we are doing exceptionally good in terms of research. So which I'm not saying this is being recognized by Australian Research Council. And recently in our Excellence Research Australia, we have done pretty well. And for the first time, we have been awarded a higher ranking in the area of industrial biotechnology. So the institute where I am, that is Flinders Health and Medical Research Institute. This is housed in College of Medicine and Public Health, and there are a few other world-leading institutes over there. And coming to biotechnology, this is what we are doing here at Flinders. We are heavily into bioprocessing. We work in the area of nutraceuticals. We are producing some pharmaceuticals from marine materials, from plants, from animals. And our major objective is to have some functional foods which can be used for various applications and that can enhance quality of life. What we are doing under industrial biotechnology, that is, we are manufacturing proteins, enzymes, carotenoids, and single cell oils. I would be talking about single cell oils soon. We use nanobiotech techniques, and we use some of the nanomaterials for enhancing all these applications. So one of the presenters, they have already introduced me, so I won't be spending time on this slide. Yep. So these are the three different areas in which my lab is working. We are manipulating microbes for single cell oil production. We are doing enzymatic hydrolysis for producing some oligosaccharides, which are having medical applications. And then we are basically purifying therapeutic proteins, which have found some therapeutic activity. That against some AIDS virus, we did some trials, and now we need to gear up what we can do for COVID. But that's an alternative discussion some other time. So the plan for the today talk is, because as the workshop is heavily focused towards biofuels and what other bioactives can be produced from microalgae, so I would be talking about single cell oils, how we can produce those oils, and how microalgae is a suitable pathway, and how we can tune the lipid profiles, how we can extract, and what way we can make the production more cost effective. Now, this is something which I wanted to show all of you, that this is what is happening at the planet. So why the single cell oils are really contemporary, that is need of the hour. So we don't want this thing to happen on the planet. So that is why, to save planet, we need to evolve some alternative strategies. And what could be those alternative strategies? This is what we thought is going to be something that we can use microbes, because I'm basically industrial biotechnology. I had been playing with microbes for the last 25 years. And in the last 12 years, we have done some work that we got some beautiful microbes. They look beautiful. They are full of oil. And then what we did, can we milk that oil from those organisms? And the answer is yes. Okay. So that is what I'm going to justify in next few slides, that how we can get those oils. And basically, oil is a lipid. All my learned colleagues, they are talking about lipids since yesterday. There was some beautiful presentations which I admired. And I would be focusing on marine ecosystem. Why? No doubt it is natural, but what we found, it is more nutritional as well. Okay, when I'm saying nutritional, it is looking after or giving us the components which are basically required to take care of the diet. I'm not saying that this is being dictated by the market which is there. So you got such a market which is dictating the production of these nutraceuticals in largest quantity. That is why the need is there to gear up and come out with such bioactives. Now, these are the different products which are available in the market, and I'm sure that all of you have seen it, to which we call as omega-3 fatty acids. This is a multi-billion-dollar market. That's why the algal area is hot. Okay, so in addition to that, there are other products which are available, but another exciting product which I'm really going to share with all of you is algal butter. So this is no longer a dream, it's a reality, and it is going to be available. Okay, and other areas in cosmetics, in cosmeceutical, some of the bioactives, they have been there. But one interesting thing which I'm going to talk here, and I would like to stress here, that the huge expansion is happening in India, where the workshop is happening. And TD, like Deakin TERI Center is in the right direction, India, China, Brazil. So there is a huge emphasis in evolving such cost-cutting technologies. Okay, so what are these omega-3 fatty acids? I'm sure that now the audience is aware of, people were talking about it. I would simply say that they are polyunsaturated fatty acid. And just for your information, the most common one is DHA, EPA, and DPA. I didn't go into the full chemical name, but if you want, the chemical name is there. docosahexaenoic, then is EPA, okay? So, but one thing which is there is the requirement. This is the requirement, right? Which is for a normal daily requirement for a human being, yeah? And that is not met, okay? The source which is there, fish oil, that is not enough. Now, how do these fatty acids they look like? Just for a basic biochemistry lesson, saturated doesn't have any double bond, monosaturated, single bond, and polyunsaturated, multiple bonds. And that is what I would be focusing in the next 10 minutes. And some of these, like saturated, they are sitting on the triglyceride portion. Some monounsaturated, they are sitting on that, and some polyunsaturated. So they are generally available in a triglyceride fraction, okay, when we look into the biochemistry. Now, this is something which is really striking, and I'm sure that this will excite you, that there are different sources of these omega-3 fatty acids, but they are not enough. Now, this is a information which was given by DSM, the biggest multinational in the area of nutritional products. And what they say, that there is a huge gap. That means in terms of the demand, which cannot be met. So that's why people have to gear up and come up with technologies which can see the day of the light. Okay. And another paper which recently appeared in Nature Food in the first issue in Jan 2020, the gap which is there, that can only be met, right, either through microalgae or through genetically modified crops. So that means what we are doing, that is in the right direction, okay? So how we can do that? I think we all are convinced with the benefits of omega-3 fatty acids, so I can skip this slide. Now, how we can produce it? So majority of the speakers, they have covered on the microalgae is a beautiful pathway of producing it, but I would be focusing more on a different type of a microalgae, which is, I'm sure that you all are aware that they are photosynthetic in nature, they can cultivate using carbon dioxide, and they can be used for producing biofuels, okay? Biofuel is basically a lipid, and these are the different species. One of the speakers talked about spirulina yesterday, Haematococcus, and decimus. Okay, but I'm going to talk about another microalgae to which we call as a heterotrophic. One of the speaker briefly mentioned about it, right? So why we are looking for this microalgae, because this grows like a bacterial system, it multiplies quickly, it doesn't need any what we call as sunlight, it grows well, and it can grow on some really cost-effective carbon sources. Now, these are the different algae which are there in the literature, and I'm sure that you all are aware of this table. This appeared in a long time back in Chessie's paper, but one thing which I would like you to see, that the organism which I'm going to talk about is Schizochytrium. Okay, and you see the oil yield, which is from 50% to 70%. So they are full of oil after Botryococcus. So they are really good organism to work with. Now, should be done to understand about the oil production. There are few key factors which needs to be looked into. How we can get a good growth. Obviously, if we are working on a phototrophic, then definitely temperature, sunlight, those are the critical components which are there. And obviously it depends on what sort of microalgal species you are working on, okay? So, but in our case, we opted for heterotrophic pathway, which is more user-friendly and more convenient to manipulate. So the first thing which we always do, bioprospecting, although it is not flavor of the month, but this is the basic thing which one has to do. We went to the different areas, we brought some samples into the lab, and these are the different locations. And mind it, there is a one location on the top right corner. This is Goa, okay? So because we had some collaboration with Indian like partners, in addition to TERI, and so we worked together on that, okay? So how do they look like? So because the title of the talk is Characterizing Microalgae, And this is the first thing which you need to look into. The first one is morphological identification, right? How does the colonies look like? Then is microscopic. You can look into the size and shape. Then you can stain it. Staining is really good. It gives you an idea in terms of the lipid content which is there in these cells. Okay. And then we can do molecular identification, which is a benchmark which in a lab one has to do. And obviously that will help you in doing phylogenetic analysis. Okay, these are some of the basic techniques which are. So these are the organisms which I was talking about. They are really beautiful organisms, full of oil, as you could see. And in one of the image, what I'm showing is that the fruiting bodies are coming out of it. Okay, so these are some of the isolates which got over here. And this is how do they look like under a simple microscope at a resolution of 40x, right? They are really illuminating microbes, which are giving you a good sense of oil. Okay, now the next step is basically what are the different stages, okay, from algae to oil. People have talked about it. They have shown a lot of data, but these are the basic things which we need to do. You have to grow, you have to ferment. And mind it, we have to disrupt algae to get different bioactives out of it. And if they are proteins or if they are unique lipids, you have to purify, then refine, and then you have a beautiful oil, which you need to stabilize as well. I noticed the earlier speaker has talked about encapsulation, so I need not to talk more about it. So obviously this oil needs to be kept what we call as stabilized, only then it is going to be more useful. Now, this is what I thought that this will give you an idea that how does algal farms look like, all right? So these are some of the images. In the center, this is a California coastline, a beautiful algal farm which is there. And this is the other one is basically it's a closed pond facility. I remember people were talking about open pond, but when we are talking about nutraceuticals, which we want to use it for consumption, human consumption, mind it, we need to have a contained environment. So open pond may not be a good option, but I won't be going into that debate, but these are the various options which could be looked into. And this is something which we do most of the time, that we grow our algae in a bioreactor. And this is how does it look like, okay? So I will show you some more images which will invite your attention. But this is what we are interested in looking into, that these are the fatty acid profile of these different type of lipids which are there in the algae. So these are the almost 25 strains which we collected, and all have different fatty acids which are there. And out of this, we are interested in DHA, which is here, omega 3 and omega 6, which is arachidonic acid, some beautiful levels which are there. And now is the time to start building on it. Okay, now this is how you will find it, that these are some interesting profiles. They are good in palmitic, they are good in stearic, they are good in DHA and EPA. Not all, but some of the cases. So you can always give it things to which I call as tuning of fermentation, okay? So for that, we need to have a good understanding of what metabolic pathways they are being obeyed in an organism. So generally there is a pathway which is known as PKS, but there is another pathway which is fatty acid synthesis pathway, okay? So we need to look into whether it is an aerobic or an anaerobic pathway which is happening, and based on that we can tweak with the metabolic setting of the organism, and then we can enhance lipid levels, okay? I won't be talking more about that what sort of stress can be provided, because it is quite established in the literature that when we give stress to the microalgae, then lipid accumulation happens. These all mechanisms are quite established, okay? But I would be talking more about the fermentation approaches which we have followed in with respect to improving the lipid content and specifically the type of lipid content which we want to increase. So some simple approaches are mutagenesis. With this, we have done some work, and there are some reports which are appearing in the literature. Some recombinant microalgae, people are manipulating it with respect to chlorella and with respect to Sandesimus, so that those microalgae, they can be manipulated for improving lipid content so that biofuels, they can be produced in a cost-effective manner. Okay, so another important aspect which is associated with lipid production is that you need to get the lipids out of it. Okay, so how that can be done? So only then we can have realization of single cell oil. So what I thought that I'm going to focus on only two things, because cell disruption, and it is going to be followed by extraction. So disruption of cell membrane and walls is desirable to improve lipid release. Okay, now we are talking about lipid is there. You need to take out the lipid out of it. Okay, so lipids are separated from other compounds by, like generally people, we use solvents to extract lipid out of it, right? So that is the only method of choice which is available. People use hexane or a chloroform methanol. We have evolved some green methods by which we can extract lipid out of it. So, but they need to be extracted and then purified, okay? So these are some of the approaches. There are some alternative solvents, or we can do enzymatic separation, or we can use nanomaterials. Right, now this is something, there is a cartoon which is just put up that lipids want to get out of the cell, but there is a rigid cell membrane. Interestingly, the membrane is not that rigid in the case of microalgae. It can easily be disrupted. People are using ultrasonication. People are using basically what we call approaches using enzymatic, but these techniques are quite lab-based. They have not been adopted at industrial scale. So still there is a huge component which needs to be developed. Okay, so basically we have done a combination of these two things, and based on that we have extracted lipids. Now another thing which I would like to invite all of you, so a young scientist in making, they are looking into it. I would strongly encourage you to look into some of the solvents which should replace chloroform, hexane, methanol. Okay, so look into some of the terpenes, look into some. I know ionic liquids are still, they are expensive, right? So, but if we can get eutectic solvent, right, which can be extracted from a plant, okay, and that can be used for extracting lipid, it is going to be a win-win situation, right? So we are also looking into some membranes which can be used for extracting lipids. That's something which we are trying here, and my student has just submitted a honors thesis. It has been graded very high. So we got some interesting stuff. We use some nanomaterials, and what they can do, that they can make some ionic interaction, and based on that we separated it. Now this is a study which I thought that I must share with all of you, because once you got the lipids, the next part is that you want to consume those omega-3 fatty acids. Okay, so what we did on the right-hand side corner of the slide, you will see that there is a dynamill. This is also known as a bead mill, okay, which is used for disrupting microalgal cell. And then we extracted the lipid, and then we used a lipase. A specific lipase was used, and that led to concentration of omega-3 fatty acid. So the concentration was increased twofold, right? So this is a good way, because in market, the omega-3 fatty acids which are available, they are basically concentrated one, but they are concentrated from the fish oil, and they do it by distillation. So what we are proposing that rather than using those methods, it is better to use enzymes and use this process for basically concentrating it. So, and coming to the story back, that these are some of the green solutions which can be done for lipid downstream processing. In summary, enzymatic terpenes. I haven't touched upon the supercritical fluid extraction. This is another a downstream technique which could be used, which people are falling in the industry. But what is happening with this? This is our what we observe, that we are not getting the lipid yields equivalent to what we can get with the solvents. Okay, so solvent is really a good option, but if we can switch towards green solvent, it will be really good. I have already shown you, but this is something which may interest you, that this is a very nice setup in which water is being treated, and then that water goes to basically open ponds to cultivate microalgae, which can be used for basic feed purposes. Now, this is Australian setup. This is I happened to visit the farm. It's close to Melbourne, and it's a beautiful farm, which and this facility is there, okay? And I'm more convinced with this facility. No doubt I love wine. I'm in a state which produces wine, but since I'm advocate of heterotrophic fermentation, and this is a setup, this could be used for growing that microalgae for producing single cell oils, which are high value and which are of multi-billion-dollar industry, right? So there are challenges which are there. I won't say that there are no challenges. I'm sure that people have been talking about it. If you're talking with respect to phototrophic, yeah, it's a light, carbon, nutrient. That's the biggest challenge. And in case of heterotrophic, what I would say that specifically with respect to carbon, that is one of the biggest challenges. Rather than growing microalgae on a glucose can be another carbon source. We have tried that, but because of interest of time, I won't be discussing it now. So I think one of the speakers spoke about spirulina, that the things are in a phase. Dunilala has been commercialized in Australia and Israel, and in US, there are many companies which are producing algal bioactive, okay? And for the information, these are some of the successful commercial ventures, which I thought that I'm going to share with all of you. So, which are producing astaxanthin, which are producing biofuels and pigments, right? So, and these are some of the commercial production which are happening using some of the heterotrophic and some of the phototrophic. So these products are available in the market, guys, and you can consume it. And this is something which we are in the process of cracking. We are working with industry partner, and you can easily see from the image which I have put up, that can give you some idea that we are working on some of the lipids that can be used for making a revolutionary product. Yeah. So what we are doing, as I said before, we are trying to create a biorefinery. Okay, biorefinery is, this word came from a petrochemical refinery in which one can produce multiple products. Okay, so we are talking about now third generation of basically biofuels, right? So biofuels are not cost effective. That's why the whole algal debate started going towards nutraceutical. Yeah. So do I have 2 more minutes? Yeah. So this is how does it look like. So biorefinery, what you can do in a biorefinery setup, from a same algal species, you can produce lipids, which could be used for PUFA. Some can give you good palmitic acid or other fatty acid that can be used for biodiesel. You can get good quantity of protein. You can get good quantity of sugars, which can be used for bioethanol. we can produce some antioxidant like astaxanthin, and then even whole biomass can be used for various applications. So this is what I always say, that this is how a biorefinery should look like. You feed the biomass, and on the other hand, you are going to get few products out of it. So we also did some work on this side. We produced two components together. We produced omega-3, and we also produced ethanol. So it is there populated in the literature, some of the work which has come from my group. And just for your information, people were talking about astaxanthin. It's a beautiful molecule. It is being used for by aquaculture industry to enhance the nutrition in fish. Okay, we did some work. We did some animal trials, and I can show you that we fed our hydrolyzed algae after extracting lipid to these zebra fish, and what we found that omega-3 content was increased. So which is a very good indication that heterotrophic algal biomass can be used for feed. Now this is a very big industry, aquaculture, right? And this has a number of applications that can be diverted to, but I think you need more time to talk on this. So probably this is my last slide. So what I would say, that nature is a beautiful reservoir from where we can interesting microalgal species. Fatty acid profiling can basically assist in better bioprospecting, because this is a back and forth. You get good fatty acid profile, you go back and you collect more microorganisms, right? That's the way. Fermentation can be tuned in order to get some really exciting fatty acid profiles, okay? And those fatty acid profiles, they can be used for preparing good functional foods. Vegetarian patty, that's another product which is available in the market, right? And I'm sure that this is going to be quite popular in India as well. Macca's, they have already come with a vegan patty in which omega-3 is there, okay? They have replaced the animal fat. Guys, this is a very big thing. Replacing animal fat is the next revolution which algae is going to make. We can use agriculture residue for economizing production, and nanobiotechnology is something which is going to be assisting us in bringing cost effectivity to the process. And if we can extract some valuable co-products, I've given you one example, which is astaxanthin. Certainly, we can hit the sweet spot that we can produce this bioactive cost effectively. Now, before I finish, now this is a video clip which my student has prepared, which I will be running soon. And I would be failing in my duties if I'm not going to thank my team. There are many collaborators and many workers here, and this is a recent group photograph which we clicked at the time of Christmas. I'm really thankful to my past PhD students and my existing PhD students and my postdocs, those who have really worked really hard to get this cracking. Yeah, thank you very much. I'm happy to take questions.
Yeah, thank you so much, Dr. Puri. And we can take the questions. So if any of the participants are having questions, so they can raise their hand and we can give them the rights, audio rights. And so it is requested from participants.
Munish, you also mentioned, I believe, that you would be running a short clip.
That's right. Yeah. So should I run it?
Yeah, I think so. You can do that.
Okay. All right. Okay. Stop sharing. Then I have to stop sharing this, and I will now share the video clip. Just bear with me. I'm just pulling the file. Yeah. Is it working?
No, not yet.
Okay, just a moment. Rishi, I have sent you a copy in parallel.
Maybe we, in the meanwhile, we can take questions.
You can now share. There is some technical glitch in the tool regarding the sharing. Now you can share it.
Okay, all right. So it will stop for a few seconds, then it will start. Okay.
No, we don't see it. I would.
Can you hear it?
We cannot see it neither, or we hear very vaguely and not really clear voice.
So actually, what I have done alone, I have sent it to Ruchi's email. She may be getting it. She can upload it.
Right. So in the meanwhile, Ruchi, I think we can take up the questions because we're getting the for the next.
Yeah.
So we have one question from Rishabh Goenka, and the question is, how can microalgae help in carbon sequestration and in climate change?
That's a very big question, actually. All right, okay. The solution which I'm proposing is basically that is in that direction, that why not to grow an algae on the available carbon sources which are cost effective in nature. And for example, there's a lot of agriculture residue which is there, and there is a tremendous forest residue. I'm talking about to which we call as waste. Okay, that has a huge carbon footprint, and that can be used for growing some of the heterotrophic microalgae, and that can help in mitigating the issues which we are experiencing.
So we are still waiting for the presentation. We have not received it. And we can have questions from the participants if they can.
Sure.
Yeah, but
I will send you again if, okay, if the first email hasn't reached you, give me a moment.
Another participant from Apurv Upreti. Question is, can microalgae help in bioremediation?
Definitely, it will, because that is what I have shown in one of the slides, that there is a treatment plant and in which microalgae is being used for remediation purposes. And so I don't work in that area, but there are a few groups, those are working on that side using microalgae for bioremediation purposes. So what happens with that, that the metal ions which are there in the water, those get accumulated in the microalgae, and then those metal ions, they can be extracted and harvested, and they can be used for other purposes. And the water is basically being remediated. So it is devoid of basically the contaminants which are there. So it's a huge area on which people are working. And I'm not working on that side, but definitely microalgae. For example, people have devised microalgae growing on carbon dioxide. I'm just giving an example, although it is not associated with remediation, but yes, microalgae can be used.
We have another question from Dr. Shawan. So I request our IT team to unmute Dr. Shawn Mundel.
I could join from here.
Yes.
Could I speak from here? Yeah.
Yeah.
So it was a fantastic presentation, sir. So actually I have a question about query about extracting the oil from algae, from the grinding method, we could extract from any kinds of seed, just like jatropha seed, they are having 40%, we could extract it. Like the mustard seed, we could extract it by grinding method. In algae, Buttercoccus burni, they are having 50% lipid, or if you could stress they are having more than 50%, could you use those kinds of technique to extract oil directly?
The answer is, you see what is happening in the industry at the moment. People are using solvents. Hexane is being used. Chloroform methanol combination that is being used. Still, it is a softer as compared to the hexane. Okay, they are in use. That is the basis of getting these pigments out, and that is how the things are coming out of it. So that is what I was saying. Industry is looking forward to some of the methods which are green, cost-effective, faster, and they can get you to better lipid yields. Okay, the answer is we have to go back to the plants. Plants has got some wonderful terpenes which we have used recently, and what we have found, they have given us the comparative yields. The only thing which you need to do is that you have to have, you need to combine treatments. Yeah, so some solvent followed by some enzymatic combination. You see what you need to do. You need to first of all break open the wall of the microalgae. Once that is open, which can be done easily by using pre-treatment, right? So with that, you open the wall, and then you can make holes, and then the other solvents which are more friendly in nature, and then they do the magic. and then they can extract the lipid out of it. So what we have seen that we are getting almost, for example, if 60% lipid is being produced in the organism, we are getting almost 55% out of it. So the answer is such methods should be adopted by the industry. And we are working with some industry partners. They are really looking forward to adopting those methods which we have evolved. So the things which have been used in the case of extracting oils from seeds, I think the mind, the thinking process is going to be changed soon, because when the algal industry is coming up with single cell oils in next 2 to 3 years time, the day is not far, okay? And then those methods would be followed by larger oil processing companies which are extracting oil from plant seeds.
So now we have another question, which is from Gauri. Can algal biomass be subjected to cold press for oil extraction?
Amrit, can you repeat it again? Sorry.
Can algal biomass be subjected to cold press for oil extraction?
Oh, that's a wonderful question. We haven't tried yet. We haven't tried, but definitely that can be looked into. What we did, which I can share with all of you, that we have tried wet biomass. We have not cold pressed it, but we have used wet biomass as such, and we tried extracting lipid out of it using environment-friendly solvents. It has worked, but the lipid yields are not to the level as you can get with the solvents which people are using. So we need to work a little more on that. But some reports are coming up in which people have used some combination. But coming back to your question, it has not been tried yet.
And another question by Dr. Sunil Pabbi, that I would like to know that lipase treatment is specific. That means it gives us exclusively the omega-3 oils. And what is the recovery percentage? Any losses? And do we have to use specific lipase?
That's right. Wonderful question. Thanks, Dr. Pabbi. So we have used a phospholipase, okay, that was specifically aimed at hydrolyzing N3 position, okay, because what we found from our TLC investigation, that what is at N1 position, N2 and N3 position of the triglyceride, once that was confirmed through NMR, and then what we did, we immobilized a specific, not immobilized, we used a specific phospholipase, and then that was incubated with the oil, and with that we could, and then we could get concentration of DHA. The recovery which we got, that was around 67%. We haven't got, I would say, more than 90% of recovery, but it was 67% at that occasion. And you need to have, interestingly, this phospholipase which we used, that was basically available commercially. And this is TL400 lipase. And then we specifically ordered this phospholipase from, I think it was, if it is on top of my head, it was a basically yeast lipase, because we looked into that what position we want, and that was a reason of selecting a specific phospholipase. So yeah, that is what we did. And we also have populated last year that then we immobilized same enzyme on a nanomaterial, and can we get the similar recovery rate? The recovery rate was similar to the soluble form of enzyme. So the answer lies in basically making those lipases more stable by using a nanomaterial, because phospholipase, as you are aware, it is quite expensive, right? It is not like a lipase, it's a phospholipase specifically working on that. So because it was expensive, that's why we immobilized it. Yeah.
Another question is from Dr. Ambati, and the question is, did you try any other enzymes for extraction methods instead of lipase? And is it economically viable?
Okay, we have tried viscozyme, we have tried nuclease, we have tried a specific protease for hydrolyzing cells, okay? And we have tried X enzyme. Why I'm saying that our no, that was with the industry partner, so I can't disclose. But that's a proteolytic enzymes which we extracted from fruit waste, and that has worked very well. And we did a pilot study which was aimed at hydrolyzing 100-gram biomass. But we haven't done a study where 1 kg of biomass could be used because we don't have a setup for doing such a big study. So we could do 100 grams and it has worked well. Rather, I can say with confidence, perhaps you will see our publications soon, okay? So that has worked very well. So that's why I'm an advocate of enzymes. I'm an advocate in terms of using plant-based terpenes. They are the way going forward.
And we have another question by Rai. As you have said, green solvents should be explored. So far, the cell disruption and extraction, is there any scope for application of biosurfactants produced from bacteria for lipid extraction?
To be honest, I haven't tried, but definitely surfactants, they will lead to better access to the membrane. This is what I personally believe. So obviously, if you're going to surfactants, they will help you in dissolving it, better access to the membrane. This is with respect to those algae which are a bit rigid, right? So because the organism on which we are working on, that's a soft organism and you can easily disrupt it. But if the cell wall is complex, okay, it's a for example, like it's a multi-chain and a really rigid cell walls, then definitely surfactants, they are going to play a good role, but that could be explored. We haven't done that. Yeah.
So we have another questions, but before that, because I have not received your video clip, so can you share the same video with Mr. Vikas, if you have his mail ID?
I can.
Mr. Puri,
Yes.
don't share your video. Instead, you can try to share it with the screen. Just share your screen and then play the video.
Sorry, say again, because?
Share the screen and then play the video.
So first of all, I should stop sharing, right?
Yeah.
No, you should share your screen and then play the video from your screen.
Yes.
So there is a share screen function at the bottom.
Yeah, look, this is what I'm trying. Yeah. Let me. Now I have to show.
It's coming. Yeah. So we can see your screen now. You can click your video.
Yeah, I'm going to do that. Yeah. And
Yeah. We're not seeing anything.
Can you see now?
Now, yes.
Okay, so then I can start it from the beginning.
Yes.
I'm sorry for that.
No problem.
No sound.
There is no sound in this.
This is at the maximum sound. I can.
I think it's good to share with Vikas Bhati.
You can share the film with the Vikas Bhati. Colleagues from the boardroom, what do you want to take forward now?
So meanwhile, we can take another question, which is, can microalgae help in cancer treatment through biotechnological means? Dr. Manish, yeah.
I think you can send those questions. Yeah. Send
So
those
we have a
questions to Manish
question if
later.
you are not.
Because he's busy in sending the clip. So
Yes, I think you share the question, yeah.
You can send the questions to him later, and then he can reply, and we can reach out to the participants with the answer.
That would be better. Yes, sir.
I have stopped the video. Yes, please tell me what you want. Yeah.
Question. Go ahead.
We had a question. If you're not
Yes,
busy
please.
in sharing the presentation, so we can have this question. So the question is, can the question is, can microalgae help in cancer treatment through biotechnological means?
Oh, okay. Right. Actually, I can't do multitasking. Just give me a moment. I'm going.
Yeah, that's why we'll share the question with you later, and then you can revert with answers.
So,
Okay. All right. Okay.
Colleagues in the boardroom, what is the next agenda?
So we have discussion in this, like after this we have to go for the discussion with the participants, I think, which is already continued.
You can open that now and for the participants for discussion.
Okay, sir.
Because
So
they're
now we
getting
are
late on the
opening
session.
so we are opening this for the discussion, and from the participants, if we have any questions, any of the participants are having any question, they can raise their hand, and we will unmute them. So, yeah, please.
Okay. So should I answer the one which was asked me? Can microalgae help in cancer treatment? Or should I leave
Most welcome, sir. Yeah.
Right. Yeah. Okay. So the astaxanthin molecule, which has been extracted by majority of the scientific community. So what we recently observed, that if you combine astaxanthin with another compound, that shows anti-cancer activity against, we have tried against breast cancer, we have tried against colon cancer, and we are trying against hepatic cancer as well. These are preliminary studies. There's not much data which is available in the literature, but The answer is there if we can extract these compounds, but what we need to do, we need to protect these compounds from the external environment. That is, that we have to encapsulate. We have to make them really make them more bioavailable so that they can give us better activity. So the answer is yes, that the compounds which can be extracted from microalgae, they can be used.
We have another question. How can algal cooking oil be manufactured from biomass? As in, how is processing different than that of SFA and UFAs?
Okay, so what is there in a cooking oil? So what sort of a cooking oil which people use? Either it's a coconut oil, or it's a canola, or it's a mustard, or it's a what we call as a basically the plant or the palm oil, right? These are the different types of oils which are available these days. So now what we can do, that is what I introduced you through tuning, that if we can increase the content of those fatty acids, then definitely we can produce cooking oil from microalgae. Okay, so you have to have a microalgae which should have a pathway which doesn't secrete polyunsaturated fatty acid. So the solution lies in some of the algae like Rhodotorula. There is a big group in Greece, and there is another group which is based in UK. They are working towards using microalgae for making cooking oils. So that was the first aspect of your question. And Amrit, can you tell me the second component, which I just forgot? What was that?
It was in how is processing different than that of SFAs and UFAs?
Yeah, so, okay. So that is what I am advocating here, that let us use solvents. Let us use basically environment-friendly solvent, because when we talk about cooking oil, we are going to be directly using it. We are directly consuming it. So if you are going to use hexane, if you are using chloroform, or if you are using acetone, yeah, so then those residues, they will become part of your metabolic activity. So use environment-friendly solvents. They will be much better than your other processes which are being used for extracting oil.
So we can now move towards the next component in our agenda, which is discussion and brainstorming session among the panelists and others on identifying challenges and strategies in algal biofuel commercialization that poses significant hurdles over the next 5 years. So we welcome all the panelists and all the participants for a brainstorming discussion on this.
IT, if you can unmute Martin, Dr. Martin, Dr. Pabbi, Dr. Pavanjatur.
In fact, I would request Hope to initiate this brainstorming discussion. Hello, am I audible?
Yes, you are audible, Dr. Pabbi, go ahead.
I just said that maybe Dr. Alok can initiate this brainstorming and maybe put forward certain points which can be discussed, you know.
See, the basic central point in this brainstorming is that there is a big gap between the industrialization of biofuel production, and we have been talking about that the biofuel production in itself may not be commercially feasible. However, it is important that we need to know how the technologies are moving forward in a biorefinery approach, in which the value addition from the other products can make this feasible. Excuse me. So our learned speakers as well as the participants are free to provide their inputs, particularly that how the biorefinery approach is moving, and if there are case study that they can refer to which are economically feasible and demonstrated clearly. As you see, there are few companies who emerged in the world scenario for manufacturing biofuels, but they have not really done very well. The only company which is still working and doing some part of the production is the heterotropic strategy to produce biofuels. So in this session, we really want to understand if there are more unknown case studies where the whole value chain is mapped on the cost economics and found to be feasible.
Hello, can you hear me, sir?
Yeah, go ahead. Go ahead.
Yes,
Yes, Rangarao.
yes. So first we need to focus on cultivation aspects. Cultivation is very, very important if we want to work towards bioactive compounds from microalgae.
There's some problem with your audio at our end, so
okay.
Trying to figure it out.
Okay, I am audible.
You're audible, yes.
Yeah.
This with some disturbance along with it.
Yeah. Okay. So see, we need to look into cultivation aspects. Either it is a photobioreactor or ratio ponds. So this is more important whether we want to reach industrial level, industrial scale up. Whether we want to work in either industrial aspect or we want to work in any biological aspects or in terms of production of metabolites from algae, either it is high value molecule or maybe a few of it can show biological activities, but the lacking is cultivation. Especially it is lacking in the why it is not successful biofuels because of cultivation. There is no biomass. That is the disadvantage microalgae. This is my experience I am sharing you. And then people are working photobioreactors. Photobioreactor is a very expensive process. So if we talk about photobioreactor, but now the people are moving into photobioreactors for production of high value compounds, for example, astaxanthin, for example of PFOS, for example, lutein or beta carotene. So this is my concern. Rather than doing a lot of things, better we need to focus on cultivation aspects. And just now, Dr. One of our speaker focused on Dr. Shawn Mundel. He discussed more about contamination aspects. So in US, most of the people focus facing the problem, same problem because of predators. Because of the predators, we cannot cultivate more, longer time. So that is the reason people are cultivating shorter time rather than going to longer time. So this is one of the concern from my side. Thank you very much for giving me opportunity to discuss with this point in front of the panel committee.
Any other insights or point of views on value chain economics?
Yes, so I'd like to add with Professor Ranga. So, yes, actually, when we talk about the industry, like what they're telling about this thing, like the biomass, what you told, like we don't have, like we can't make like this real amount of biomass, and those things for me, like in my personal opinion, algal biofuel that will be like be successful when we think this algae as a crop, like the regular crop, like that's when you are growing like this rice, like in the field, we can think in that way. We do not need like a very skilled labor on that thing. So we need to develop some robust technology where I could grow microalgae in the field. So that's the research effort, like we need to have that one, because if we can't scale up those things, because like you see like there are several industries they started, but they cannot reach in that scale. Like they reached up to pilot scale, not like the real industrial scale. We need that scale actually. That is the most challenging in the microalgal research.
Okay. Any other point of view?
So,
Pawan, you
yeah,
want
this is Juto.
Yeah, go ahead.
So, so one thing where I think we have to look forward in scaling up process, especially because we are talking about biomass. Definitely, it is a constraint for the huge production what we expect for biofuels. But there should be a way where, because my assumption or hypothesis here is, can we extract these high value products which are forming as like bit into the media, or can like called a milking kind of process, where you are not destroying the cell. Instead of not destroying the cell, you can milk out. See, there are certain ways, like you can extract the omega-3 fatty acids into dodecan layer, and that kind of technology. So basically what we are doing is, whatever the cell is able to produce, if we are able to capture or extract it through that means, by just milking out that particular product, I think that will be one of the best assets to include in the cost economics, because this is where very less, or I can say very minimal work has been done regarding the milking of omegas or anything kind of thing, which is very reasonably a very good cost effective way if you can do it. And also we are actually so there are some classical examples, because in couple of talks, even Reliance Life Sciences in India are doing a great work in terms of microalgal cultivation, because I have seen in one of their talks that they were able to cultivate the seed culture almost for 1 week. That means 4 different seasons or 3 different seasons. So that means it is possible to get biomass, but the thing is, are we able to once we are getting the biomass, and to reduce the doubling time, that is to capture how fast it can be, because these are all photosynthetic, so you cannot pressurize the system to work at a minimal level and go to the maximum. So that's what, whenever the system is able to produce some high value product, can we sequester or can we extract that by a simple way without destructing a cell? That will be one kind of economical way of looking into that. And other perspective is like, because we want algae to grow at one stretch for 20 days and give us a product. I think that is not what is we have to look at an industrial perspective, because when you see microalgal growth, initially if you can get biomass quicker in terms of whatever experimental pattern it is, if you can approach 2-stage method, so that will be a very good, for example, you increase the biomass and give it a stress, because within 24 hours to 12 hours to 48 hours, it can give double the amount of lipids. So you have the biomass, you have the lipids in 2-stage, then again, you can extract those cells and recultivate these cells. And why I'm telling is, in my lecture also I focus like, you can replete the cells back into the media, into normal conditions, and you can attain as a normal cells as it is. So there is a possibility how you can, as much as cost economics can work it out. And these are main lacunas, that's why we are not able to scale it up. So if we can work out on these areas, I think that also will be an asset what we are talking about. Thank you.
Thank you, Pavan. Addition to what you said, there's another challenge which is looking pretty much clear to all those who are in this area, is the fractionation and separation of high quality products. We do talk about multiple products available from algae, but we still have to do a great deal of job in terms of separation and then producing high quality product, single products or multiple products from those pool of products that are available within the biomass. Dr. Pabi, you wanted to say something?
Yes,
Uh,
actually, may I continue?
Yeah, go ahead.
Okay, as mentioned by Dr. Alok in the beginning, that there are very few or very few successful ventures for biofuel. Of course, there are many options, and many of these have been tried and tested, and the economics has also been worked out. But the economics which has been worked out for a particular organism or a particular set of conditions may not hold good for all other places or conditions. So because this economics can only act as a kind of a, I would say, benchmark or something like that. But then for every kind of set of process, these economics will be changing. So these need to be worked out in a particular set of conditions. And what, in my view, we need to have an integrated, of course, it is well said, but what I say is that integrating biofuel production with especially the CO2 sequestration, because both of these are basically addressing the environmental issues when we talk about petroleum products and about the CO2 level which are increasing. And it has been seen that there are many strains which can grow very fast under high CO2 concentrations. So we need to look for those high producing strains. And secondly, we also need to look for the quality of lipids, as Dr. Pavan has been referring, because lipid production, higher lipid production can be there in many strains, but the biofuel like lipids, that is what we are interested in. And secondly, when we talk about our country, we have many seasonal variations. And so instead of going for what we call as a one strain, can we have a what we call as seasonal rotational concept, where we have maybe a couple of strains or maybe 3 strains which can be grown throughout the year as a rotation, so that the whole process is I would say viable throughout the year-round production kind of a thing is there, because such kind of large-scale productions are more feasible in open, because we have a lot of sunlight in our country. So in Indian context, these are the options which I feel need to be addressed in a much better way. Thank you.
Thank you, Dr. Pavi. Munish, you wanted to say something?
I think I agree with what you initiated, Alok. Biorefinery is the best way going forward. And another approach which I would recommend is using heterotrophic microalgae, right? And tune it according to the type of valuables you want to produce. You can use some strains for producing triglycerides, which are going to be good for biofuel, okay? And you cannot say that the technology are not scalable. Yes, technologies are scalable. People are already producing it. And there are examples in Europe, there are examples in the US, which people are doing it. It is only that what we need to do is let us list what valuables, which are high value, we can produce through microalgae pathway, and that is going to bring cost effectivity. And I know downstream processing is a big challenge, but not to the extent as we have that challenge with respect to lignocellulosic biofuel. Mind it, algae is a much cleaner system versus plants. Like that is what my personal feeling. And we have tried different type of lignocellulosic biomass. They are really, I'm sorry to say that they are dirty as compared to algae. Algae is a much cleaner system to work with and much delicate, and it can give you more yields in a lesser span of time. And with context to India, as Dr. Pabbi said, that there is a good sunlight which is available. So heterotrophic microorganisms, they are good in terms of producing high lipid values, okay, but you need to grow some other phototrophic microalgae. For example, they are giving good quantity of lutein, Hematococcus, that's another algae which give you very good astaxanthin yield, but we need to evolve a process by which we can extract astaxanthin. At the moment, the market for astaxanthin is hot. Astaxanthin is available for $5,000 a kg. I'm talking about international market, right? So, and there are plenty of buyers, aquaculture industry, they are looking forward to getting that product because Hematococcus need to be grown for good 20 days. So can we have that Hematococcus produced from another microalgae by manipulating? I'm referring to a non-GM approach, right? So by non-GM approach, that can be used for benefiting human food, and then the leftover biomass can be used for feed. In India, there is a huge industry. We have hatcheries and animal feed where that leftover biomass can be taken up. Thank you.
Thank you. Thank you very much. Is there any other suggestion, point of view? If not, I think we are now coming to. Yeah,
so we
go ahead.
we have received the video clip from Dr. Munish, and we can play it right now.
Okay, we can do that because time is running. It is 2:05 p.m.
Wide range of applications from nutraceutical supplements to third-generation biofuels. Further exploitation of microalgae as a source of valuable fatty acids is a very important step in the eventual transition into more renewable industrial practices for lipid processing. The production and extraction process consists of a complex multi-step methodology to get from the initial algal biomass to the eventual oily extract, which I will take you through now. Generally, the production of lipids from microalgal biomass has 6 major steps. These are culture growth, freeze-drying, cell disruption, lipid extraction, purification, and refining. Growth of the microalgal biomass can be performed using a variety of different media and culture conditions, depending on the species and strain of microalgae being used. This produces a substantial quality of wet biomass containing intracellular lipid ready for extraction. Unfortunately, due to the high hydrophobicity of most common extracting solvents, lipid extraction from wet biomass generally has low efficiency. As a result, we freeze-dry biomass to remove moisture to overcome this issue and significantly improve lipid extraction efficiency. While lipid extraction can be performed directly after using the dry biomass produced, it is favorable to further improve extraction efficiency by applying a cell disruption step. Cell disruption procedures are divided into 4 major classes. These are physical, thermal, chemical, and biological. These are highly interchangeable, although some may be more suitable for specific species of microalgae. This allows the breakdown of cell walls to improve lipid availability, without which there may be a substantial loss of lipids. Lipid extraction is generally performed using one of 3 different solvent systems. These are the Falk, Bly-and-Dyer, or Soxhlet methods. All 3 of these involve the use of hydrocarbon solvents to facilitate the transition of lipids out of cells and concentrate them in the extract. Due to the toxicity of most common solvents, more environmentally friendly solvents are now being considered. Although the best existing systems still involve either hexane or the pairing of methanol and chloroform. The raw lipid extract can be put through numerous analytical tests to determine overall yield and individual fatty acid quantities, which we will discuss in more detail later. Individual fatty acids can be separated from the overall mixture by purification to allow concentration of lipid segments such as omega-3 and polyunsaturated fatty acids for nutraceutical supplements or saturated fatty acids for biofuel production. These separated lipids are then put through a standard refining process to improve color, clarity, and smell, and remove impurities. This will produce a pure fatty acid concentrate which can be used for commercial processes. We will now be considering the most important of these steps in more detail. The main stages of lipid extraction are split into two categories which are often analysed and optimised independently in the laboratory. These are upstream processing, which focuses on the accumulation of biomass and lipid, and downstream processing, which entails the extraction and analysis of fatty acids. Both are vital to receiving large yields of valuable fatty acids. Efficient upstream processing is required for collecting a large quantity of biomass with a high lipid ratio, while efficient downstream processing is required to receive the majority of fatty acids without significant product loss. The biomass growth and lipid extraction stages are the most manipulable in the laboratory to enhance desirable fatty acids in the final extract. Growth may be manipulated through application of different culture conditions and media compositions. Lipid extraction may be manipulated using different solvents, solvent ratios, and extraction conditions. Here we will focus on the upstream processing stages of biomass growth, harvesting, and lyophilization as we conduct them on thrust-chytrid biomass at the Flinders University Department of Medical Biotechnology. Then moving on to downstream processing, we will go through an efficient method of lipid extraction, followed by the respective analyses of the lipid extract generated. The aim of the culturing process is to accumulate a large volume of biomass with a high proportion of intracellular lipid. Popular microalgae frequently used for lipid extraction include chlorella, Hamidococcus, and Schendesmus. At Flinders, we utilise various strains of thrust-chytrids due to their exceptional lipid content and high proportion of omega-3 fatty acids. These microalgae can be grown in smaller volumes in earthen flasks or greater volumes of industrial significance using a bioreactor. The minimum growth time of a microalgal culture is 7 days, of which 2 days should be allocated for generation of an inoculum and at least 5 further days for development of biomass in the main culture. We use a simple growth media called GYP media, with 3 major components dissolved in artificial seawater. These components are glucose, yeast extract, and peptone, which together provide all the essential elements for the growth and development of microalgal cells. The artificial seawater is prepared using instant ocean salts from aquarium systems in a 50% concentration. Use of a simple GYP media is useful in that it allows for easy preparation and manipulation of conditions and components in the lab when we're attempting to enhance the volume of biomass produced or the proportion of lipid accumulated. We prepare inoculum with 5 grams per litre of glucose and 2 grams per litre of both yeast extract and peptone dissolved in 50% artificial seawater. We prepare 100% artificial seawater using 33 grams per litre of instant ocean salts in distilled water. To our media, we add 2 loops of thrust-chytrid subculture, then incubate the inoculum for 2 days on an orbital shaker at 25 degrees and 150 rpm. After the 2-day period, we prepare the main culture using this inoculum. The main culture media is also a GYP medium, but with adjusted nutrient concentrations. We prepare this using 10 grams per litre of glucose and 1 gram per litre of both yeast extract and peptone, also dissolved in 50% artificial seawater. We inoculate this using our inoculum in a 5% concentration, which is the equivalent of about 50 ml of inoculum for 1 litre of medium. This is also incubated on an orbital shaker at 25 degrees and 150 rpm. The maximum amount of biomass occurs after 3 days, but lipid accumulation continues to increase up to between 5 and 7 days. Hence, harvesting is conducted only after a minimum of 5 days. Upon harvesting, microalgal cultures are transferred to appropriate centrifuge tubes, which should be pre-weighed to allow later determination of the mass of biomass accumulated. Biomass is centrifuged for 10 minutes at 4,500 rpm to separate the microalgal cells from the media. The supernatant is then removed and discarded, and the pellet is stored in the -20 freezer until further use. In converting the wet biomass to dry biomass, the biomass should be transferred to a -80-degree freezer and stored for at least an hour to ensure the biomass is sufficiently frozen for freeze drying. Freeze drying is performed at -80 degrees and 100 millibar using the frozen biomass to allow the removal of moisture. This is left for a minimum of 24 hours to ensure that the biomass is properly dried. The centrifuge tube with dried biomass should then be weighed to determine the mass of dry biomass produced by subtracting the initial empty tube weight from the final weight. Dry biomass allows significantly improved lipid extraction efficiency compared to wet biomass and therefore is frequently used in the laboratory and industry for high lipid yields. However, the eventual development of better extraction procedures is necessary due to the high energy input required in the freeze-drying process, especially if microalgae are to be exploited more widely for commercial fatty acid production. The most important part of processing lipids from microalgae is the lipid extraction process itself. While this can be performed using virtually any amount of microalgal biomass, generally we use 10 milligrams of biomass for experimental purposes to improve ease of handling, allow higher throughput testing, and minimize solvent use. However, it must be remembered that when using very large proportions of biomass, such as 10 grams or above, lipid extraction efficiency may be slightly reduced. As mentioned previously, the major lipid extraction methodologies are the Falk, Bly and Dyer, or Soxhlet methods. We utilize an adaptation of the Bly and Dyer method in our lab using either hexane or a combination of methanol and chloroform. The dual solvent system of methanol and chloroform is the highest efficiency lipid extraction system available and is therefore most frequently used in a laboratory setting. However, hexane is more frequently used in industry due to improved safety for products intended for human consumption. In addition to this, while having significantly lower extraction efficiency than the methanol and chloroform system, hexane remains the best single solvent system available. If hexane is the chosen solvent, 600 microliters is added to the dry biomass under an appropriate fume hood. If methanol and chloroform were chosen, 200 microliters of methanol and 400 microliters of chloroform are added to the dry biomass, also under fume hood. The amount of solvent used can be easily adapted to different amounts of biomass, but the most important factor in doing so is ensuring methanol and chloroform remain in a 1:2 ratio, as this has been found to allow highly efficient lipid transfer. Regardless of the solvent choice, the dry biomass and solvent should be mixed sufficiently by vortex for at least 2 minutes, and then centrifuge for 10 minutes at 10,000 RPM. The supernatant should be removed and retained, and then the addition of solvent repeated a further 2 times for a total of 3 extractions from the same biomass overall. The total supernatant is then filtered using a 22 micrometer hydrophobic filter into a pre-weighed glass jar. Finally, the lipid extract should be dried in a heat block under a fume hood at 55 degrees for 3 hours to ensure that no solvent remains. For larger amounts of solvent, evaporation using nitrogen gas or a rhodovapor may also be applied. The lipid extract can then undergo lipid analysis. Lipid analysis can be performed in a number of ways. We usually utilize gravimetric analysis to determine overall lipid yield as a percentage of the original biomass. We then use fatty acid methyl ester gas chromatography to determine the fatty acid profile, which shows us the individual proportions of the different fatty acids present as a percentage of total fatty acids. Once both the initial weight of the vial and the final weight of the vial following lipid extraction have been obtained, this can be used to determine total lipid yield. The weight of recovered lipid is equal to the subtraction of the initial weight from the final weight. This is presented as a proportion of the amount of biomass extracted from to demonstrate how much lipid can potentially be gained from any weight of dry biomass. To do this, we simply divide the weight of the recovered lipid from the weight of dry biomass used, allowing us to show lipid yield as a percentage of dry cell weight. Using fructochitrids, we have generally been able to achieve lipid yields of between 25 and 50% dry cell weight without application of cell disruption treatments. FAME-GC analysis may then be performed on the lipid extract. This should be performed as soon as possible after extraction to prevent oxidation of polyunsaturated fatty acids, with storage at -20 recommended until FAME-GC can be performed. The lipid extract should be dissolved in toluene to allow the conversion to FAMEs. To this we add BHT solution, internal standard, and acidic methanol. The BHT is an antioxidant solution which reduces oxidation of PUFAs during the conversion process. The internal standard is used as a standard to assist calibration of the gas chromatograph machine. Acidic methanol is the primary agent for the conversion of fatty acids to FAMEs and must be prepared fresh an hour in advance. The reaction is incubated overnight at 50 degrees in a cap tube while the conversion to FAMEs proceeds. The next day, the FAMEs undergo a cleaning procedure to remove impurities which may affect detection by the gas chromatograph. This begins with the addition of a 5% sodium chloride solution. This is then transferred into a separate test tube for extraction of FAMEs into hexanes. The hexane layer is removed and transferred to another fresh test tube where potassium bicarbonate is added to wash the solution. The top hexane layer is again transferred to a separate test tube where sodium sulfate is added to dry any remaining moisture. The FAME mixture can then be transferred to appropriate containers for FAME GC analysis. The gas chromatograph gives an output displaying the varying amounts of each individual fatty acid relative to each other, and each is presented as a percentage of total fatty acids. In our lab, the thruster cultures have been found to be highly versatile, producing a significant amount of omega-3 PUFAs and also a high amount of SFAs for application in both nutraceutical supplements and biofuel production. Newer and more environmentally friendly techniques are now becoming a bigger focus in lipid processing, with a significant desire emerging for greener, more environmentally friendly technologies to aid high efficiency lipid extraction without producing large amounts of toxic and hazardous contaminants or high energy consumption. The discussed methods are all highly transferable and facilitate high throughput experimentation on accumulating lipids from microalgal biomass. Together, they constitute well-established procedure for processing lipids from microalgae and are extremely useful to researchers in the lipid processing field as a foundation for optimizing and developing new and exciting green procedures. I hope you found this talk on processing lipids from microalgae to be both interesting and informative. Thank you for listening.
So I would like to extend my deep appreciation to Dr. Munish Puri for managing his busy schedule and joining us for sharing his ideas on microalgae based research problems, and also pointing out the technology for single cell oil production and various methods for downstreaming processing of omega-3 fatty acids. Now we'll move towards the closer of this workshop, and I would like to invite Dr. Alok to take this workshop to a close with his concluding remarks. Please, yeah.
Thank you, Ruchi. As we mark the end of the two-day workshop on the high-end characterization of microalgae, overcoming the technological barriers, I would like to take this opportunity to recognize the role played by our speakers, Dr. Pavan Jatur, Dr. Sunil Pabbi, Dr. Nirupama Malik, Dr. Martin Lopez Garcia, Dr. Amritpreet Kaur Minhas, Dr. Shawn Mundel, Dr. Rangarao, and Dr. Munish Puri, to share their knowledge on various aspects of microalgae in this workshop. I hope that we can still rely on you for continued support as we put the different mechanism of networking into gear. I wish to commend you all for having made time out of your busy schedule to attend this important workshop. Ladies and gentlemen, it is no secret that the success of this workshop is your active participation. We are very grateful to you for taking time off from your work to attend the workshop. I must say, regardless of background and experience, participants were on a level playing ground, and each one's opinion received the attention or criticism it well deserved. So thank you for participating with us. Let us all go away from here with one mind that the best way to solve common problem is to work together. With these few words, ladies and gentlemen, it is an honor and pleasant duty for me to declare the official workshop closed. I would like to appreciate congratulations to and the support provided by my colleagues, Dr. Amritpreet Kaur Minhas, Dr. Ruchi Agrawal, Mr. Vikas, Mr. Rahul, Mr. Satendra, and entire IT team for uninterrupted and high quality management of the event. Thanks to large number of students who participated in this event, besides the faculties and researchers from all over the world. Our experiment to conduct online workshop with sharing protocols online seems worked well. However, we shall look forward for your observation and comments. See you sometime soon, maybe on yet another event. Thank you and namaste.
Thank you, Alok.
Thank you.
So may I request all the speakers to switch on their camera so that we can have a group photo together. Our IT team will be giving you the rights to turn on the camera. So we'll wait for 2 minutes and then we'll click the photo.
Thank you.